Hello, welcome to Dive 702 of ROV SuBastian. We're just getting started on our descent. We're going to be going down to about 3,090 meters or so. Probably keep it muted for that. I'll pin some comments, but yeah, let's see what we find in the midwater.
Hi everyone, all our viewers online. Welcome back to Dive 702 with ROV SuBastian. We're on board research vessel Falcor 2 on one of the southern seamounts in the Nazca Ridge. We're in the area that intersects with the seamounts from the Salas y Gomez Ridge. We're about 900 nautical miles west of Chile and probably about 1,000 miles south from Peru. We've been out here for something like 3 weeks, and we've had a series of ROV dives, and we've mapped a lot of new ocean floor on these seamounts that are largely unexplored, unmapped, and have never been visually observed before these dives that we've done. With me here in Mission Control are Dr. Aaron Easton from University of Texas Rio Grande Valley, Dr. Javier Seyanes from Universidad Católica del Norte, and the rest of our biology team. We have Claudia and Mari, and we have also Cameron from our JEPCO mapping team. The ROV is now flown by Ben and Tyler. We have Alex and Juliana managing the stream and the data and the navigation of the vehicle. My name is Tomer. I'm the co-chief scientist of this expedition. And we have on Zoom our remote participant, co-chief scientist, Dr. Jodica Vermani, who is also the CEO of Schmidt Ocean Institute. And I've been honored to have her as co-chief on this project, and we're fortunate to have her on the call with us. Hi Jodica, how are you doing?
Hello, hi Toma, hi everyone. It's my honor that you're my co-chief scientist, I think, Toma.
Thanks.
It's been quite a few weeks.
Yes, absolutely. Yeah, we've been through a lot, but I think we did pretty well, and it's been a very steep learning curve and growing experience for sure for all of us.
It's also great to have Erin there and Javier and everyone who's on board as well, who joined us for this particular cruise at relatively short notice.
Yeah, thank you. It's been an honor to be invited back, but yeah, I think we had 10 and 11 days from the time we were invited to the time we flew out to come on this expedition, so it's been a whirlwind for us, but absolutely amazing and quite an honor to be invited back for a third expedition this year. And we've been really happy to support both of you in your project goals and objectives for this expedition.
So before we went live, we were talking about how many days that's been for you, Erin, on Falcor 2 this year.
Yeah, I've been, I think it'll be 118, possibly 119 days that I've been on the ship, including the time in port. And there's 4 other team members that are at nearly that many days. I think 3 are about a week less, and then another one is 3 weeks less time on the ship because they just did half of the second expedition, or they spent less time in port on the ship. So yeah, it's been my second home this year. I think I've spent about half the year on the ship, so a little over maybe.
Wow.
Yeah, I think I've met almost every crew member between the expeditions. I know all like 6 Alexes, even Alexes that don't go by Alex.
Yeah, we've been extremely fortunate to have our collaborators from Chile and from the U.S. Return on such short notice to be part of this expedition. We've benefited a lot from their very intimate knowledge of this region and their really deep understanding of, no pun intended, but their deep understanding of the ecosystem here that we're trying to understand.
Yeah, and I think the insights we gained from being on the past expeditions in terms of selecting the dive sites and what we're learning about these seamounts has helped inform like Tomer in making his decisions as well for this expedition. So we started learning a lot about where we are finding the greatest biodiversity on different ridges around the seamount relative to the summit. So I think that's been really informative for this expedition and helped us obtain data. We keep finding more new registrations for the area. The last 2 dives, we saw a fish we had not seen before in this area, and we're able to collect it on one of those 2 dives. So each and every dive, we still have a new observation, new record, new depth, potential new species. It's amazing the discoveries that we've been making.
So, Erin, question for you: with all the data that's been collected over the past few months, how long do you anticipate it will take to work through all of it? It's a substantial amount of data, isn't it?
Yeah, it's a lot of data. So we are trying to seek funding to do some workshops so that we can bring taxonomic experts to accelerate the process of species descriptions. But just going through all the samples, so we have a sample log spreadsheet here, and I was just looking at it. We have 4,600 and some rows of information for the samples we've collected on these 3 expeditions, and so that includes subsamples. But when we go back to the lab, each of those samples is opened and looked at, and then any other associated organisms are counted and noted because we don't have time to do that on board. So our total number of specimens collected will go up drastically, and that's something that Maxi Tapiaguera has been working on. He was working on that the couple months between the cruises, updating the species IDs. Yeah, we're already working on some of the short notes and reports. But yeah, it's going to take us years to go through all the data. But some of the preliminary data is forming the basis of theses for different students, both in Chile and in the US, and collaborator institutions around the world. So that will help expedite the process as well with the students working on it for their thesis projects.
That's great. And I think we'll work with Ocean Census on getting workshops and additional taxonomic assistance there. I see there's a map on the live stream, on the dive stream there, or a positioning. Would you want to talk about that?
Do you want to take that, Tomer?
Sure, yeah, I can talk about the map. So we can see these little flags. Those are our planned waypoints. They're spaced about 250 meters apart. That gives us a reference of about 1 hour per waypoint. So we can kind of track our progress and try to stay on top of our plan. And we're starting, our first waypoint is just below 3,000. It's about 3,060 meters. And our final waypoint, which we might not get to because it's pretty far, is just over 2,000. And our track, I planned this track that kind of climbs on this ridge line that kind of zigzags towards the summit of this knoll that is separate a little bit from the main seamount that we've designated as T11 in our project. It's T11 is a large guyot has volcanic origins, of course, from long ago when the Nazca plate hotspot was still active. And it used to have a reef on it, a carbonate reef on it, that was at some point exposed above the sea level and was eroded to a flat top. And then at a later phase, some of that has likely collapsed. The southeastern flank of that volcanic cone has probably collapsed and left a big scar. And we are actually diving on a knoll that kind of lies to the north of this guyot, but connected to it, still part of the same feature. And in our dives here in this region, we've kind of noted that most of the times we get a lot more diversity on a secondary feature that is lower than the main summit, or deeper and maybe a little smaller than the main seamount. Not sure yet why exactly that is, but we can think about it. And therefore we plan this dive to try to intercept some of those deeper habitats, that deeper biodiversity. And this map was made with our multi-beams, our echo sounders that are mounted on the ship on Falcor 2. And we have 3 of them, and we usually use 2 out of the 3 and these depths. We have the EM124 and the EM712. They're both very similar in the way they operate, but in different frequency bands, and they kind of adapted to different depths. So when we travel with the ship, the multi-beams kind of project a very specific waveform, an acoustic waveform at 12 kilohertz in these depths, and that creates a fan-shaped beam that kind of sweeps or scans along the seafloor as the ship moves forward. And we're at a depth of about 3,000 meters. We can cover a swath of, let's say, maybe 8 kilometers or so, or 9. And we were able to create a 3D model of the seafloor with depths about 15 meters apart. So we'll get a 15-meter grid of depth of soundings. And that's how we recreate the seafloor in this 3D model that Alex is showing us on the screen right now. And that's what we use to identify these bathymetric features and this kind of terrain that will hopefully have the highest biodiversity. So it looks like we've just touched bottom. At 3,080 according to ROV SuBastian's depth sensor. And we're right on top waypoint 1, and here we go. We're starting this this traverse. So a lot of our viewers have been following us on this expedition, and we've had 12 or 13 dives, and of course on many previous expeditions as well. we recognize a lot of these names. And for those who are new or not familiar yet with what we're doing, we are operating a remotely operated vehicle that has lights and cameras and 2 manipulators that are highly articulated. They have, I think, 7 joints or 7 functions, and they're operated with a special kind of joystick by our ROV pilots here in Mission Control. And we use those arms, those manipulators, to collect samples and bring them back on board and analyze them in the lab. And that's most of the scientific work that takes place on board. It takes a big team to do it. It takes a long time to be out here on the ship. An expedition is usually around a month long, give or take. And here in Mission Control we have a dive leader who is one of the lead scientists, and we have a person who logs the data or the scientific observations and samples, and we have an ROV pilot and an ROV co-pilot. When the ROV pilot actually flies the vehicle and the ROV co-pilot operates the different payloads that are on the vehicle. We also have a multimedia technician who makes sure you're getting the stream loud and clear and bright, and with all these cool overlays and different camera views. We also have a marine technician who takes care of all the different navigation systems and data streams coming from the vehicle. And that's just inside Mission Control. Outside Mission Control, we have an entire ship. Our ship, Falcor 2, is 110 meters long. I don't know how many tons that is, but it's a lot. And we have a crew of about 35, I believe.
In terms of from top to bottom, it's what, something like 7 stories, 7 floors?
Yeah. Yep. 7 decks, you could say.
Yeah, 7.
It's a lot of stairs.
So you get. Yes.
Right. They put the
So
gym in
there's
the
a
best
question.
place.
Oh, sorry.
No, I was joking that they put the gym in the best place, because then when you're done, you have to climb all the way up, all the stairs back up to your cabin to take a shower.
You can get lasers on that. These are huge.
So I have a question. There was a question in the chat from Emma about the whale beaks that were found, and is there an update on there? So maybe as we are exploring here, Erin, do you want to talk about what happens to things like that?
Yeah, so when we first bring it up, we take all the living organisms off of it and preserve them separately. And then we dry the fossil out. Like all the other samples that we're collecting, or most of the samples that we're collecting on this expedition and the past expeditions, we take them back to the Universidad Católica del Norte, to their Sala de Colecciones, so their museum and aquarium there, and they have a collections, and all the keep going. All the collections are logged into an international database so that others can request access to them in the future. The samples then are processed there, and can you look at that really quick? Thanks. And so some of the samples are going to end up going to the National History Museum in Chile for samples that were collected within the national jurisdiction of Chile. Those outside, we have more freedom to send them to other collections internationally, so that some representatives, if we collect 2 or more of an organism, or in this case we have multiple fossils, so we have the possibility of sending them out. So yeah, if we can sit down and zoom, I think we're going to collect this. So yeah, that's what we do in terms of the fossils. We need to find collaborators to do additional work with them. All of us on board are biologists, so we need to find like a geologist or someone who works with fossils to see if they want to do any additional analyses on them.
Darren, I see where
yeah, you broke up a little bit there, but I'm trying to see exactly what this is. It looks like it's a coral, but it might be, yeah, they're going to get situated a little bit closer so we can get a better zoom on this. See if it's a coral or maybe a sea pen. It definitely has an ophiuroid hanging out on it, so we'll probably do a collection here because I don't think it's one we've seen before. We don't generally dive this deep on that we haven't generally dove this deep on prior expeditions, so we are expecting to see different fauna here than we have observed and collected before.
Pilots are now going to land the ROV gently on the bottom to try to not stir up the sediment. It's very fine. And they're going to position the vehicle in a way that allows the co-pilot to operate the manipulator and retrieve a sample.
Yeah, so one thing that we like to do is try and collect a representative fauna that we think are potential new species or new reports for the area. That way, a lot of times to have a confident species assignment, you need to have the sample in hand. There's some where you can see the diagnostic characteristics in video alone, but if it's a potential new species, you have to have a sample in hand really to do a proper description. And for some taxa, you actually need to have 2 or 3 or 4 or 5 individuals to describe a species. So there's different standards for each taxon. So sometimes we have to collect a second or third or fourth or fifth specimen so that we can do a proper species description. Yeah, can you zoom in on it first? I'm not actually sure yet what we're looking at. Yeah. Thanks.
I haven't added up all the numbers yet, but on the 12 or 13 dives that we've done on this expedition, we've had at least, I believe, 2 dozen
2 dozen samples per collections per. Yeah, I think this dive this morning we had our fewest number at only 7 collections.
Right.
But generally we were having around 2 dozen collections per. I think our greatest collection was almost nearing 30, 28 or so.
I think we had a dive with 31.
31 was it? Okay.
Yeah.
Knew it was up there.
Yep.
Yeah. And those are just the actual individual collections. A lot of times we end up with more samples when we process them. Some we separate right away. Like the other day we collected a coral that had crinoids on it, and we pulled the crinoids out and put them in a separate jar. But all the fluoroids that were on it we left on there, and we'll sort those back in the lab. And so those were just assigned 1 sample number for now.
So we're taking a pretty close look at this coral.
Yeah.
Does this look like anything you've seen before?
It's not something I've personally seen before. It's not something we've collected before. I'm not seeing the diagnostic characters really well here, but let's go ahead and try and collect it, and then see the lighting with a light background. It's hard to focus in on the diagnostic characters, especially with it being a mostly opaque, transparent sample here. So we're going to try and do a collection. I would grab it from probably near the base there. I'm not sure if it's something that's going to retract or not. And I don't have Maxie in the room. He is our resident expert on all fauna in the area. He has done his undergraduate research, his master's thesis research, and now he's doing his PhD research on the fauna of the area. So he really knows all the fauna better than anyone else. He has an incredible mind for remembering diagnostic characters. So
grab the sample. There's several different types of containers for them depending on the type, size, and shape. So we have these elongated tubes with a lid that they're called the coral quivers, exactly for samples like this. And then for bigger things we have a bio box, which is basically a big box with a lid that we can open and close hydraulically. We also have a suction sampler that we use to handle very small or delicate creatures, and those are just kind of vacuumed into a carousel with jars, and we can rotate the carousel. So every time a jar gets full, we switch it over to the next. We also have 2 rock boxes that are basically like a milk crate for our geological samples. We have the ability to also sample sediment cores with a push core. We also have Niskin bottles to sample water. We keep track exactly of each sample, take a picture of it, and note where it was taken and which container it is put into.
Thank you.
All of our containers are mounted on the front porch of the vehicle. The porch can be moved, it can slide in and out on hydraulic rail. You can now see it being stowed for flight. We have several cameras on SuBastian. The main science camera that we use, the one that you see the most, the one that's actually on the screen right now, is the 4K science cam, SciCam. It's basically a Sony AX100 in a housing that's rated to 4,500 meters with a zoom lens. And it has native 4K resolution. Other cameras that we have. Oh, go ahead.
Oh, no. First of all, I was going to say nice job to the ROV team. But also I was going to actually, I think you were going to talk about it. I was going to, maybe we could talk about some of the new technologies that have been tested and the new sampling capabilities on this cruise as well.
Yeah, we've had a couple of really cool developments for this cruise that we deployed for the first time and tested, and they were very successful. One of them is what we call Jason's jelly jars, which is basically, they're named after our engineer Jason who designed them. And basically the jelly jars are special because they're designed to sample the gelatinous creatures that we normally see kind of hovering in the water column, the pelagics. They don't have any bones or any physical structure that can support them, and you can't grab them with a manipulator, and you can't suck them in with a vacuum, with a suction sampler. So the only way to really get hold of them is to put them inside a big jar. And basically those jars are like a vertical tube with lids on the top and the bottom that we can open and then fly the vehicle and kind of navigate it to get the whatever gelatinous creature it is, for example, a jellyfish, get it inside the tube and then operate the lids to the closed position. And we've successfully done that with a jellyfish on the first dive that we did on this expedition. And yeah, it proved itself. And then another cool system that we have is the selfie camera and the selfie stick that you can see right now on the screen. And what you can see is basically an HD camera that's mounted on a stick that is deployed from the port side of the vehicle. It rotates out until the camera faces the face or the front of the vehicle, which you can see right now. And that shows you both of the manipulators, the starboard and the port manipulators in their stowed position, and the front of the ports. You can see those white tubes. Those are the coral quivers, and behind them are the bio boxes, and above that is the main camera, the 4K science camera. And what's cool about the selfie stick is that it gives us a brand new perspective on those close encounters that we have with everything, the fauna and the flora, everything. We're in front of a big sponge forest or a big coral, or just hovering very close to a tiny ctenophore or siphonophore, or you immediately get a beautiful perspective and kind of the relative sizes of everything, because SuBastian is quite a large vehicle, so it's always interesting to see how it compares and how it relates to the environment.
Yeah, I've really enjoyed seeing that, the relative size of creatures relative to SuBastian, and then having the other cameras show close-ups. It's been really, really interesting.
Yeah, we've been enjoying it a lot. It took us some time to kind of tweak the location of the camera and the different lights on the vehicle. There's also a light on the selfie stick itself, and we had to design and produce with our 3D printers different kinds of hoods and filters and diffusers to get the lighting just right, to have enough light from the selfie camera and enough light on the object that we're filming, but to not drown out the selfie camera, not blind it. So it took a few kind of rounds of trial and error and kind of tweaking it, and I think we brought it to a pretty good place that kind of proved the concept.
Yeah, definitely. And then, of course, the other new thing we tested was this telepresence capability.
Yeah.
As I tune in from about 5,000 miles away.
Yes, we've had a number of collaborators on shore, and Jodica, of course, you were the first one to test it and to be on one of the previous dives. I forget which one it was. But how's the experience from shore?
Oh, it's great. Yeah, it's very easy to tune in. I think now that we've got this set up for Falcor 2, we had it previously on Falcor, but now, you know, this is a different mechanism and a different stream, but it's super smooth, super easy to use when we have that connectivity with the vessel. So I think this now opens up for many scientists from around the world to take part in these expeditions.
Yeah, I also think it's nice that, you know, we can bring in experts and have these conversations, right? And so it's more informative for the fans and the public out there watching and rewatching the streams. So it's not just us pointing at what we're seeing, but an expert can come on and tell you a lot more than any one scientist on the ship might be able to tell you about a particular organism or environment. So I think it has a lot of promise and it's really been exciting for us to participate in this trial that you've been doing, and I'm interested in seeing where it'll go from here on future expeditions.
Yeah, I agree, Erin. Absolutely. And maybe you'll join us virtually in the future.
Maybe. That would be great.
Aaron, aren't you supporting remotely one of the next ones?
I am, yeah. Yeah, I'm a co-PI on an expedition that's a two-leg expedition, October-November, but I'm not planning to be on board for a number of reasons. Mostly I teach a lot in the fall, so, but then I can participate more readily remotely, I think, with this new tech that's been tried out.
And I did also enjoy having our oxygen minimum zone experts join us, because that was in cruise we didn't have an ROV dive for, because it's looking at oxygen levels in the water column. So having them come on board and explain remotely, that was wonderful, Toma.
Yeah, that was really great, especially because everything we see here, this entire ecosystem is controlled by that oxygen minimum zone. It's one of the big, what's the word, constraints, or one of the top-down controls on this area. So it was really eye-opening, I would say, to learn about the significance of the oxygen and how it's measured. Actually, the concentrations are so low that they had to design a whole new technology just to be able to detect that trace amount of oxygen.
And we have their device on it again now, the M-tail, right?
Yeah, the M-tail is mounted on the ROV on SuBastian, and while we're not getting the live measurements from it because it has to be calibrated after the dive, we are logging all the data. So every time the ROV comes back on board, we download the data and send it back to the lab for calibration. And that's going to be, I believe, part of the data set of this expedition, of course, and part of the research that will be done with all of this data that we're collecting. We've also had Larry Mayer.
Yeah, I was going to say that was great too, with
Yeah.
mapping expertise.
Yeah, it was really special for me, at least, to speak with Larry, as he used to be a professor of mine and also my boss for a number of years when I worked at the University of New Hampshire. So, yeah, we were fortunate to have a little bit of his time as he was just flying out to way northern Greenland to be on an icebreaker for 2 months. And he talked about the significance of mapping the ocean floor and how the shape of the seafloor is even related to things like climate change and sea level rise. So it's been fascinating. The possibilities that we have now with the remote participants are endless.
Well, we will definitely get more now that we've opened this channel.
Yes.
So as we continue to explore, I have a question for you and Erin, and actually anyone else. Javier, I think you said, was in the room too. What's this is the last dive of this particular expedition. So, what's been your highlight so far?
Well, for me, the highlight was definitely seeing that extensive and ancient coral and sponge forest. By my estimates, like rough estimates during the dive, it went on for some 800 or more meters of us transecting through it, and there were definitely corals and sponges that were centuries, if not millennia, old. And I counted at least 17 genera of corals and 4 or 5 of sponges. And so we had even more species than that. And so of all the dives we've done this year, I think we saw the greatest diversity of corals and sponges in one location with among the highest density of ancient corals that we'd seen as well. So that was really a standout for me on this particular expedition. We were also impressed with another seamount not too far from there that had quite high biodiversity in the oxygen minimum zone, much higher than we're used to seeing. And so those were two of the highlights for me.
It was pretty spectacular seeing that. I think that was dive 698, right?
Yeah, dive
Handful of dives
698.
ago.
Yeah, and I mean, I think rough estimate we were seeing 3 to 4 meter long Rhodean iridogorgias and iridogorgia and bamboo whips, and then 2 to 3 meter wide coral fans of different genera. It was just really impressive.
Jodica,
Fantastic.
what was your highlight?
Oh, good. Well, remotely, I think when you sent me the images of the before and after mapping of the
Yeah.
newly discovered seamount, that was such a contrast. It was incredible.
Yeah.
I don't know if that's your highlight too, Toma, but those 2 images were brilliant.
Yeah, I agree. That was definitely one of the most exciting discoveries for me on this one.
Yeah, and I remember correctly, it was a pretty interesting dive too. I'm really glad we went back there. Like we basically passed that, and we're like, well, let's go and map over it. Something's there. Let's go see what it looks like and dive on it. So yeah, good decision.
Yeah, it really shows how unknown this part of the world is, and almost every rock we turn here is, we find something new. Every patch of seafloor that we insonify with our sounders is new.
Yeah, that's what it was, right? We actually crossed over it on our way to another seamount and had it mostly mapped, and we went back to finish mapping it and dive on it. That's what it was.
Yeah.
Things get a little blended together when you're diving, mapping. You have to look back at your notes to remember the order and which dive was which.
And what about Javier? Oh, speaking of rocks, I see you approaching some.
Yeah, we got a nice little crinoid hanging out on this rock outcrop here. Yeah. Great, thanks.
I think another highlight was the Casper octopus. Was basically the first animal we saw on the very first dive.
Yeah.
The moment we touched the bottom.
Yeah, that was pretty cool. It was just
Yep.
right there. Hey, here I am. You've started your expedition. It's going to be amazing.
Yeah.
It's like a sign.
I definitely took it as a good omen.
I think you guys have had a few inquisitive creatures that have just hung out.
Yeah, we've definitely seen a number of octopus on this one. A couple of Dumbos that were really nice to film. because they fly kind of funny with the ears, quote unquote. And we were lucky also to find a couple more of the chanocops, which was a new species that was discovered back in February, I believe. January.
Yeah, and of course we've seen on an awful lot of our dives our shauna shops,
Yeah.
which has become a little bit of a mascot of the of our expeditions this year. So we see them. We saw them on our first expedition this year, and keep seeing them.
Actually, I think we probably have the world's only stuffed chana, right? Yeah, that was
Yeah.
handmade in Chile by
Yeah.
a friend of Maxi.
Yeah, crocheted stuff. Got the big one and 3 small ones. Each department has their own. Actually, Shoreside should have their own small one too. So,
Right.
yeah, it's been hanging out in Mission Control all expedition.
It's definitely the mascot.
Here, like the Pathopathes, the black coral. This one looks different than what we see shallower, so we're gonna go ahead and collect it.
Maybe after this collection, you can ask Javier what his highlight's been. Oh, that's okay. I didn't know if he
Yeah.
was in the room still.
Yeah, no, he stepped out. I don't know if he's taking a break since he's off shift.
Yeah, this is gonna be a long dive, and 3 a.m. the biology team rotates out. They have 2 shifts, so I think Javier is on the other shift, so he probably has to be here at 3 a.m.
Yeah, and the other shift had the entirety of the last dive. We went in the water. Well, I guess we were in the water just before the end of our shift. Yeah. So, but we were descending, so we didn't reach bottom until our shift ended. So, and then the bulk of this dive is on our shift. So it's worked out pretty well actually that way, where we've been able to get some break time in between, even though we're a smaller team than we normally would work with for 24-hour ops. It's worked out quite well with the timing of the dives and everything, and the mapping in between to keep us well rested and excited. And people will get up a little off shift to help out. It's been a lot of teamwork. And, wow, great images here. Can really see that fine detail. All right, great. We can go ahead and collect it and then do a camera shot, see if we can get some more of these amazing images with the polyps and tentacles. Yeah, this is a black coral, so it'll be, how big is it? Do we want to put it in a quiver, or is it too big? Yeah, I think we're going to take the whole thing because, yeah, we'll put it in a quad then. Thanks.
I think another important aspect of this expedition that maybe not all of our viewers are aware is that it's entirely in international waters, in the high seas, in a region that is kind of vulnerable because there's no law protecting it.
Yeah, and it's an area that, from the few studies that have been done here, have shown that it has high endemicity, or a number of the fauna are reported only for the region. So they're unique to the region. It has high biodiversity, high uniqueness, and so it's been identified as an ecological and biological significant area, and it's one of the top priorities for protection on the high seas. So yeah, any of the information we're collecting here can help inform its hopefully potential future protection. So yeah, we're collecting a black coral here, and black corals have a very tough skeletal structure, and so they're very difficult to cut. So we're going to just go ahead and take the entire colony in this case, because chances are if we try to take just part of it, we'll take the whole colony anyway. So if we take it from near the base, then it'll come up in good condition. And again, this is looks like a morphology of a black coral that we haven't seen in this area. I'm not sure if it's a known species or not at this point. We'll have to do more work up in the lab. Corals are notoriously difficult to identify the species from video data alone.
What kind of work do you do on it in order to identify it?
Yeah, so there's we do a lot of genetic work and then microscopy work. So I work more with octocorals, and they have these small skeletal elements in them called sclerites. And so those are one of the diagnostic characters. And sometimes when we're doing max zooms, you can kind of see the overall form or shape of the sclerites, but you have to actually look generally microscopically. We use a scanning electron microscope typically to look at them. And so requires us to collect them and remove the tissue and then look at the skeletal elements. So, and then genetic work, of course, will help us identify them down to genetic groupings. But general morphological characters we can see, like Bathopathes, this is the genus. It's kind of this center skeleton, and then it's got these paired branches coming off on the side.
Yeah.
So, yeah, and these are excellent images of the polyps and tentacles. So the black corals are hexacorals, so they have 6 tentacles per polyp. And so you can kind of see the set of 6 there. So keep seeing, like it's like 1, 2, 3, and then 1, 2, 3. So those set of 6.
Where does one find a scanning electron microscope?
Well, nowadays you can get benchtop ones for about $100K. So
No kidding.
yeah, $100,000 to have it on the benchtop, and supposedly you can take the one I have. Supposedly you can take it in the field. They're like tried telling me like, oh, you can take it on a ship, but it's like a 200-pound piece of equipment, and I don't know. Maybe it could be set up on a ship, but I have like an anti-vibration table because vibration's not great. So yeah, this is excellent. You can go ahead and store that when you're ready. But yeah, there's probably at least 4 to 6. I looked at least 4 different companies and compared them, and then picked one that I thought was user-friendly. I could do most of the maintenance right there in my lab instead of having to ship it back to the company to do maintenance. So yeah. And then you need a sputter coater to go with it. So a sputter coater is what you use to coat the skeletal elements in gold or gold palladium, maybe. There's different metals you can choose so that the electrons are actually will interact with that, so you can actually see the details. So if you don't have them coated, then they're just like bright white. So, for the low price of $100K, right? For scientific equipment, it's not too bad. I mean, my light microscope is $70K, and so to have a scanning electron microscope that was only $100K isn't too bad. Right? Yeah. Like I could have had the additional features I wanted with a light microscope would have made it over $100K, and I couldn't afford it with the funding I had to buy equipment in my lab. So yeah, scientific equipment is not cheap. I think the sputter coater was another 15 or 20K, and they can be considerably more than that as well. Yeah, sounds good. Yeah, it should kind of hold up. Sorry, it's bigger than I thought it was, but I don't know when we'll see another one. But yeah, I actually had this summer, I had 2 high school students that I mentored, and I trained them how to use the scanning electron microscope, and in just 2 months they were able to each process 6 corals and obtain images and then do image analysis, measuring the size and width of the different skeletal elements. So.
Why are they called black coral?
Because their skeleton is black.
Why is it black?
Good question. It is, so a lot of corals have these calcium carbonate skeletons, and so they tend to be this whitish color. But black corals have like chitin in their skeleton, and so it's this dark brownish color that looks black. When you actually put it like under a light microscope, it often will be more of an amberish color, like a dark amber color. Yeah, we can go ahead and continue. Thanks. Sorry. Just gonna log this sample. There's a stalked crinoid that we saw in the background. If we just give it a quick zoom on our way, that's fine.
Which one's that?
This right here. Yeah, that yellow thing. We can just give it a quick zoom on the way, that'd be great. It's a flyover zoom.
Hey, Toma.
Yeah.
So you were saying earlier this area is volcanic, right?
Yes, I mean, largely.
So because it,
Yeah.
yeah, I'm just looking at these rocks, and it's like they almost look like lava.
Yeah, they almost look like lava. Sometimes, for me, it's hard to distinguish between the lava and just the rocks that could be carbonate, but are coated with the manganese, the patina. A lot of times
Right.
it looks similar, but yeah, I would assume these are volcanic. We can definitely take one that if we see a loose one, we can take it.
Right.
Yeah.
Actually, someone in the-
lava
Oh.
flow.
I was
But
just going
again-
to say, someone in the chat said pillow basalt.
Yeah, I'm not great with my geology either, but yeah, these look kind of like pillow lava flow to me. Yeah, there's some different glass sponges here. We're seeing at least 2 different types here, 2 different species. Yeah, we can get the zoom with that.
Yeah, with some of these rocks, we have to measure their alpha radiation when we get them on board. We have a Geiger counter.
Como la árbol. Javier, yo te quiero preguntar qué era tu highlight de la expedición. Geotica, no.
Pero ahora.
Sí, ella está. ¿Quieres?
Oh, okay.
Javier came back in the room, Geotica, so he's gonna come over, I think, and tell you about his highlight.
Hello.
Hello.
Jotica. Hello. How are you?
Yes. Good. How are you, Javier?
Yeah. Having a good time here. As always.
Excellent. I was just, we were talking about this is the last dive of this
Yeah.
expedition,
Mhm.
and it's that you've been on board again. Thank you very much.
No.
But what's been a highlight for you?
Okay. No, thank you very much for the invitation, and it's always a pleasure to be here on board. And highlight maybe we work with those gastropods of the genus Profundiconus. Maybe you don't know, but they are very special kind of gastropods that are venomous. They produce a kind of toxin that we are studying together with colleagues in Spain. And we had for the Salas-y-Gomez Ridge, but only one from here. We have another one that we have found in this expedition. So they are very rare, very difficult to find alive. So every time you find one of those, you treasure it. So I would say that this is one of my highlights, but because probably Erin already told about those ancient corals of gardens of corals, but maybe from the taxonomic group that I work, I would say that this is one of the highlights, that they have a very complex toxin that now also some bio applications that are being studied. So it's a really interesting group. We have described a new species from specimens collected in previous cruises now, and we have another new one that we are going to start describing pretty soon. I would say that.
That's great.
Mm-hmm.
Fantastic. Thanks for sharing.
Okay. Okay. Well, I will be watching the rest of the dive. Now I'm going to have dinner and probably then trying to catch some sleep because my watch starts 3 a.m.
Oh yeah. Well, enjoy dinner and sleep, and I think we'll hear from you later then.
Okay. Thank you very much, Jotica. Pleasure
Bye.
to talk with you. Bye-bye.
Bye. Likewise.
Mhm.
So again, we're just seeing some more sponges, and we saw some crinoids, stalked and unstalked. It looks like we're seeing some coral skeletons, some rubble there. They're quite large, so I'm hoping that as we continue up, we'll see some ancient colonies as we did the other day. That would be great. as Javier mentioned, we're coming up on the dinner hour, which means in about 30 minutes we all shift. So the other half of the team can go and eat dinner. Tomer's back with us.
Whoa.
Wow.
That's pretty cool.
That is pretty
Jorica, you seen that?
I am.
I've never seen.
Yeah, I think it's an anemone, and I
What?
think it might be settled on the stalk of something.
That's
Right?
that's what it's like.
Yeah. I think it might be on like a old sponge stalk maybe, and there went the dust cloud. Very fine sediment. It's coming. I think this is maybe Libonema. You know what I mean? They're always so graceful looking. So yeah, we made it to waypoint 2 within an hour. We actually had to go a little faster today, but we're making our typical pace, at least to start with, so that's good. Alright, looks like it's starting to clear out the dust. Sandstorm. So yeah, one thing that they've been trying out on this expedition is different lightings. They've changed the layout of their lights on the ROV. So, and then they've been adding the diffusers. continue to do that. Looks like there's a smaller one down below on the stalk.
Uh-huh.
This looks like something like a little pom-pom, like little kid would want to have around. Great. Yeah, jester hat. Yeah, it's lovely. Nice shadow too with the lighting as it is right now. Alright. Alright, you can take the glamour shots you want and then we can head on the way. Yeah, we don't do a lot of collections of anemones because a number of them disarm themselves. And even before they come up, they'll be in, like they'll have released all their tentacles, and so they're not in good condition to work with when they come up. Now it does. Yeah. We might be heading in the same direction of the current, so orients itself to the current. Yeah, I think we're kind of heading along the edge of this ridge right here. It's quite steep right here. See some animal tracks. Always one of the hardest parts of the dive is when we're over a lot of sediment, because a lot of the organisms are kind of transparent in color, and so it takes a lot of concentration to be looking at the screen and seeing when you can see something that might be of interest.
have a pretty nice sunset out there.
Oh yeah.
Yep.
Ooh, nice. Yeah.
Yeah, in mission control, it's hard to know what time of day it is, if it's even day.
Yeah. It's nice.
But
When I was out there earlier, it was all rainy and cloudy. Nice that the sun came back out.
yeah,
Hey, Emma, so how long is this dive? I think we've got some new people who've joined, so maybe a quick recap.
yeah, absolutely. This dive is gonna be a total of about 14 hours, and we're gonna be recovering tomorrow morning 6 a.m. local time, UTC -5. And as soon as we get the vehicle back on deck, we have to start making way back towards Chile. It's gonna be about a 4-day transit. But this dive started about 4 hours ago, and we touched bottom at about 3,000 meters, a little deeper than 3,000, and we're making our way up this ridge. This portion is covered with sediment, but we had to climb up a nice basalt cliff, or a pillow lava kind of face. So we're seeing a mix of rocky features and some sediment, and we've seen some interesting corals so far, and a pretty cool anemone, and a little bit of fish. And we're gonna be climbing about 1,000 meters more vertically over the next 4 kilometers or so. So certainly there's gonna be some steep terrain and some mixed morphology. And for those who are just joining and are not really familiar with what we're doing, we're on research vessel Falcor 2 that's operated by Schmidt Ocean Institute. And right now we've deployed remotely operated vehicle SuBastian, ROV SuBastian, that is equipped with several cameras and a lot of powerful floodlights, and 2 manipulator arms that we use to retrieve samples from the seafloor. And this is dive number 702 of this vehicle, and it's the 13th and concluding dive for this expedition to the Nazca high seas and seamounts. And we're currently about 900 miles west of Chile, and we've been out here for about 3 weeks, and we've explored a good number of these seamounts and guyots. They're pretty large features. Their base is at about 4,000 meters, and some of them reach over 200 meters, so almost kind of touch the surface. And we've been fortunate. We've explored, I think, about 10 or 11 seamounts that have never been mapped or seen before with the human eye. So almost everything that we see, and including you, our viewers, almost everything that we see we're the first humans ever to see it. And one of the things that we found was actually a whole entire seamount that was not known before. And we've made a very high detailed map of it, and we actually were able to explore it with ROV and found an amazing coral and sponge garden on it. So that was a huge surprise. about a week ago. So thank you all for joining us tonight for another dive. We have about 12 hours to go until we have to be back on deck. My name is Tomer. I'm the co-chief scientist, and on Zoom we have the other co-chief scientist, Dr. Jodika Vermani, who is also the CEO of Schmidt Ocean Institute. And here in Mission Control we have Dr. Aaron Easton, and we have also Marie-Angel Gallardo, which is a biologist with Universidad Católica del Norte. We have our ROV pilots, Ben and Tyler, and we have Alex, who is our multimedia technician and managing the stream and all the audio and the video that you're hearing and seeing. And we have very sensitive microphones too.
So Toma, the image that we just saw of what the surface view over the helideck that was shown just a few minutes ago, it looks like the weather has improved since this dive started. I think at the very beginning it looked a lot darker and grayer and cloudier.
Yeah, I noticed that. It was looking kind of dreary when we launched, and it definitely cleared up a little bit, and it's looking like a beautiful sunset out there. We've actually had a couple of pretty bad storms that we've kind of endured, that had seas of up to 5 meters and winds up to 35 knots. Being in the South Pacific this time of year is it's interesting, an adventure.
Gosh.
There's a pretty neat sea cucumber, yeah.
So, in addition to the interesting oxygen minimum zone of the Eastern Pacific, this area actually has upwelling, and with upwelling, that's currents that moves water up from the deep sea, come nutrients, and so alongside those nutrients, there is a very robust fisheries off the coasts of South America. There was a question in the chat, so I thought I'd just answer that.
Oh,
I'm sorry. Like, so amused by the sea cucumber, like doing backflips and forward bends.
Yeah. I see it now.
Yeah, a little bit of a delay. All right, I think we're good. Thank you.
The dancing sea cucumber.
Yeah. I'm sorry. Yeah, this area is really, you're right. This area is quite unique. We started out on the Nazca Ridge, and we are finishing up on the Salas y Gomez Ridge, near the junction of the two ridges. And right in this general area is where, yeah, you have an oxygen minimum zone that's pretty intense. And as you move further west, that oxygen minimum zone becomes less intense because its source is really that upwelling and nutrient production that's occurring closer to the continent. But what you get is lower nutrient conditions as you move west, and higher nutrient conditions in the area that we're in. And so we believe that contributes to some of the uniqueness of the fauna on each of the seamounts along these ridges. And they have some different geomorphology as well, where some are quite shallow and come up into shallow enough depths that they they were erosion acted on them, so that they're these nice flat tabletops. But then others were never quite so shallow, and so they're more a traditional seamount cone shape. And so there's a nice mix of those seamounts of tabletop and traditional shaped seamounts in the area. So it's interesting geomorphologically as well as for the biodiversity and the environmental conditions as well.
Yeah, speaking of the fishing that you mentioned, Jodica, we've seen some abandoned fishing gear on a couple of our dives. Some remains of lines and traps.
Yeah, didn't we have a fishing vessel? We saw a shackle on one of the early dives, and a fishing vessel actually passed us.
Right.
Right on one of the early dives too.
Right.
Gosh, it feels like that was a while ago, but it really wasn't.
Many sunsets and mission control.
I start now thinking, because I go on shift at 3 p.m., I wake up and my first meal of the day is lunch. And so the last few days I've been getting a little confused because other people are on different shift change times, like noon, 6 or 6. And I think Javier did it the other day too. He was like, yeah, after lunch. He's like, but we already had lunch. He's like, yeah, after lunch. He's like, but we had lunch. He's like, oh, yeah, after dinner. And then I was talking to someone like, oh yeah, this meal. And they're like, wait, we already had that meal. So we all get a little time warped in terms of what our meals are.
Yeah. Yeah. It's sometimes it's like feels like Groundhog Day.
But like the days are amazing. It's just
Yeah.
you get a little confused on what day it is. Is it a weekend or not? We have like signs of what day of the week it is, like ice cream Sunday.
When there's pizza, you know it's Sunday.
Yeah. Yeah, little things like that.
When there's safety training, you know it's Saturday.
Yeah, but that's just for the crew. We don't.
Right.
Yeah, so that helps you all.
Right.
Looks like rain in the distance there.
Yeah, it could be. Sometimes we get these squalls.
Yeah.
Yeah, we've been having a lot of that where it'll be like beautiful and sunny, and I'll go out to the outdoor lounge and go start my workout, and halfway through my workout it's raining. So yeah, it's been really interesting in that way on this particular expedition. It's winter here, so we've actually, in my opinion, we've actually had really good weather for winter in the South Pacific. It's known for having like high seas and like what they call angry seas basically in Chile, the marejadas, and yeah, so about once a week we're having a storm event come in. Can we sit down and go ahead and collect both of these two urchins? Thanks. So yeah, the fact that we've only had a couple weather days really where we haven't been able to do operations has been quite good in my personal opinion for the times I've worked in the South Pacific in this area. Yo creo que sí. Algo parecido. Yeah, we're just gonna collect these urchins. I don't know if we have them, but there's two in the right here. So if they end up being something different, then having two is always a good idea in case it ends up being a potential new species. I'm not sure. We'll have to send the images to an expert for the group. Yeah, we can put it.
Erin, can you?
Go ahead.
Sorry, say there's a question of what makes the bioturbation and cone mounts on the floor.
Sorry, I had to reposition my headset. But yeah, the bioturbation and cone mounts. So we find sometimes it's shrimp actually are doing some of that bioturbation. Maybe some of we saw a squatty earlier, so I'm wondering if some of the holes or the burrows are being made by the squatties. On a lot of the dives, we've seen squatties taking advantage of little rock caves, but there are some that actually, I think, make their own burrows. And so that might be something. A lot of times when you see the trails, those tend to be sea cucumber or urchin trails. But I think the cone mounds right now, without seeing who's making it, is very possible it could be a shrimp, because you'll often see them like they'll make the mound, but then they'll be in like a shallow area behind it or just tucked behind it, altering the currents. But there could be other organisms that make them as well.
Thanks.
Mm-hmm. In the sit cam, you can see the sea star mark.
Oh yeah. Yeah. You see it? Sea star mark.
You can't see it, unfortunately, in the science cam, but in the sit cam that's looking kind of more down, where they can see the porch and the cores, we can see like this, as if a sea star had either been there laying on top, or maybe it's even burrowed underneath the sediment. You can kind of see its mark it left behind, kind of like a snow angel, except for it's in like the perfect shape of a sea star. So as we move on, we should keep our eyes looking at the sediment and see if we see more marks like that. We think that maybe these urchins are in Spitodiadema. Yeah, so on our one of the earlier expeditions we participated in this year, we had an echinoderm expert on board. So a lot of times we send her images, and she will probably be the person who does a lot of work with these specimens, but we'll probably also reach out to other experts with the group, so that we have more people working on them at future workshops, so we can bring all that expertise together and process more samples in a shorter period of time. Yeah, they'll probably want to release them too, so just grab whichever ones you can with just the edge of the minute, probably. Yeah, some of these urchins can really move.
It's making a run for it.
It is. They're pretty. They can be pretty fast, actually. So yeah, we actually had a sea star make an escape out of the bio box on one of our dives recently, and we came up and we're like, "Where's the sea star?" And we went back and looked at the SICCAM images that are taken throughout the dive, and I was watching how it moved. It was in a quad bio box with 4 sections in it. It moved from the one we put it in to the neighbor one, which had a different sea star in it, and then it moved to the one behind, and it was on its way to the next one. We still don't know exactly where, like when it got out or how it got out, but it was on the move. I mean, in 20 minutes it was able to change from one quad section to another, so the opposite side of the other quad section. Yeah, they like to break off. These are never the easiest in the world to collect. Don't worry about a glamour shot, just go straight in. We'll get good images in the lab with the macro setup. You might want to just put it in a quad because the spines are going to be hard to battle to get it in.
Yeah.
Okay. Such energy, Mike.
What up, Mike?
So, where are you going, Sonny?
Yeah, it's right there. Yep, that's it right there. So we're just doing a shift change here right now. We have Mike and Sunny coming in for Ben and Tyler. Thank you, Ben and Tyler.
Great job.
Shodia says great job.
Speaking of a shift change, I'm actually going to be bowing out in a minute or two as well.
Yes, same here. Thank you, Jodica.
Thank you all for allowing me to join Thank you, Toma, Erin, science team, the ROV team, and science department on board, as well as the entire crew on board. It's been really fun watching remotely and participating remotely. So thanks for that.
Thank you, Jodica. We wouldn't have been able to do it without your support and your guidance, even though you're not on board.
Yeah, thank you so much. It's really, again, it's been an honor and a privilege for our whole team to come back and support your expedition and to be able to receive all these samples to complement our prior expedition. So we really appreciate it, and thank you so much.
All right. Thank you. Bye,
Thank
everyone.
you.
I'll be
Bye.
watching.
Yeah, I see.
Bye,
thank you.
Jodica. Hope you get better soon.
Thanks. Bye.
Ciao.
Before we go and collect that, can we zoom in on Sea Star? Because I think we're going to sit on it otherwise. Oh, it's a little sun. Okay, perfect. All right. Nice. All right, and I'm also changing out as well. So Maxie's going to be coming in for the next 2 hours. I'm going to go off and have some dinner. But yeah, we're just going to add this urchin into BioBox 3A with the other one. Thank you.
How are you today? Hi Mike, hi Sonia, hi Juliana, hi Mary. And hello everyone, Maxi here. I hope you're all doing well and welcome to the last dive. And before starting my shift, I'd like to remind you that the main goal of this campaign is to explore the seamounts of the Nazca Ridge in the international waters, aiming to explore remote and uncharted places to provide information to protect this fragile and remote ecosystem.
Want to stop these in 3A? Yeah, that's what they're, glamour shot?
Yeah, please. Always welcome glamour shot. Okay, this dive began at the depth of more than 3,000 meters, and our goal is to reach the summit or the upper slope of that seamount to 200 meters. And during this dive, we observe a homogeneous and with complex landscape with some urchin and different type of coral, for example, batypates. In short, or in summary, the previous dive in this campaign, it's important to highlight that it was one of the most spectacular dive. Was the 697 was the most spectacular dive because we observed a great diversity of coral, maybe more than 20 species of corals and more than 6 species of sponge, different types of snails and squid lobster. And also during the first dive in the dive 690, we observed the first footage of Casper octopus in this part of the Pacific. And also I will also like to thank each one of you for joining us during the dive. And it has been a fantastic expedition full of discoveries. I am very happy to be part of this expedition since it was always my childhood dream. The Nazca Reach is the most important place in that part of the Pacific because this chain hold on many secrets waiting to be discovered. That is the reason that we need to protect this place of the part of the Pacific. Hola a todos, ¿cómo están? Maxi aquí, espero que todos estén bien y bienvenidos a lo que nos corresponde a nuestra última inmersión. Antes de comenzar mi guardia, me gustaría recordar que el principal objetivo de la presente campaña es explorar los montes submarinos de la cordillera de Nazca en aguas internacionales con el objetivo de explorar zonas remotas e inexploradas y proveer información para proteger estos prístinos sectores. Como señalaba anteriormente, el presente dive o la presente inmersión comenzó a más de 3.000 metros de profundidad y nuestro objetivo es llegar hacia ir escalando hasta la pendiente o la ladera del presente monte submarino aproximadamente a las 2.000 metros de profundidad en un guyot el cual no tiene nombre y se le ha dado una asignación de número T11. Resumiendo todo lo que hemos visto durante las presentes campañas y presentes inmersiones de la campaña correspondiente a este mes, es importante destacar que durante el dive 697 fue donde observamos la mayor diversidad de corales, más de 20 especies de corales, más de 6 especies de esponjas, una gran diversidad de langostinos o squatis, una gran diversidad de moluscos, lo cual nos recuerda por qué debemos proteger estos prístinos sectores. Asimismo, al principio de nuestra aventura en la presente campaña, en el dive 690, pudimos observar el primer reporte y primera observación in situ de un pulpo Casper, o pulpo gasparín que sería en español, a 4.500 metros de profundidad, siendo el primer registro para esta parte del Pacífico. Asimismo, en la inmersión que tuvimos ayer, pudimos observar distintos nódulos, distintos tipos de ambientes heterogéneos y complejos, principalmente cubiertos por un tipo de costra que pueden ser costras de cobalto. Me gustaría también agradecer a cada uno de ustedes por estar siempre con nosotros durante estas inmersiones y que sean parte de esta fantástica expedición, la cual ha estado llena de descubrimientos, por lo cual también estoy muy feliz de ser partícipe de estas expediciones, la cual siempre ha sido un sueño de mi niñez. Y por último, decir o señalar que la cordillera de Nazca es un lugar que guarda muchos secretos que aún esperan por ser descubiertos y es por ello que es necesario proteger esta zona de futuras presiones antrópicas. puede que sean pueden que sean quizá algunos espadangoides, erizos regulares o estrellas paxilócidas quizás que estén enterradas, porque se veía un trail de una paxilócida. Now we are observing a large pycnogonid or sea spider, genus Colossendae. Thank you, Mike. Thank you, Sonny.
You're welcome. Squatty.
Squattler.
Can you open?
I can just zoom in only a quick zoom of that, and after that keep going. Now we are serving a spiny squatluster. Might say that the genus of that squatluster is Galacantha. Bueno, como comentaba, hace un par de minutos observamos un picnogónido o araña marina del género Colossendae. Una de las características principales de esta araña es que carece de tipo de mandíbulas y tiene, como se llama, estructuras más parecidas a los quelicerados, que son principalmente los pedicellarios. Curiosidades de estos organismos es que son bastante grandes. De hecho, a eso lleva su nombre de Colossendae o Colossendidae, de la familia Colossendidae. Y uno de los aspectos alimentarios que tiene este grupo es principalmente alimentarse de las Venus atrapamoscas o de sifonóforos, los cuales están cercanos al suelo, tales como los dientes de león de profundidad o termopalia, o algún batifisa o algún tipo de spaghetti monster que puede estar en la zona. Asimismo, hace un par de minutos vimos un langostino espinoso. Mari nos decía que es del género Galacantha, el cual hemos visto en gran parte de esta expedición. Ya creo que en más de 3 dives, en más de 3 inmersiones hemos visto a este pequeño amigo. The screen is landing. Now we're serving an armored shrimp, Glyphocangon shrimp. Yeah, no problem. Yeah, sorry. Few minutes ago, I told about the diet conduct of the sea spider, that the sea spider eating a different type of cnidarian, for example, the Venus flying crab, and also different type of siphonophores. That is okay. Siphonophore? Yeah. Between this siphonophore, we can find, for example, the dandelion, the benthic dandelion, thermopalia, and also Batifisa conifera. I would like to remind you that maybe in the dive 693 we saw another Batifisa conifera or flying spaghetti monster. That is the second report of this part of the Pacific. And the first one was in the Salas y Gomez Ridge, close to the Rapa Nui Island. Como comentaba, también para recordar que dentro de nuestras observaciones destacadas pudimos observar distintos organismos, los cuales destacan por su belleza o su extrañeza, y uno de ellos estuvo el monstruo de espagueti volador, por eso señalamos el Flying Spaghetti Monster. Este sifonóforo del género Batifisa, posiblemente especie Batifisa conífera, que fue el segundo reporte, la segunda observación in situ realizada en la presente campaña en la cordillera de Nazca, siendo el primer registro principalmente en la cordillera de Salas y Gómez, cercano a la isla de Rapa Nui.
¿Podemos mover unos
Yeah. Okay, can you zoom in on that version? Now we're serving a sea cucumber, the genus Amperima. Yeah, sorry, that word is very difficult to say for me. Siphonophore.
Look away. He's shy.
Oh.
Oh, pobrecito. Hizo popó. Se hizo popó del susto.
Ok, ahora estamos viendo un pepino del género Amperima, caracterizados principalmente por pertenecer al orden Elpídida, principalmente conocidos como estos los pepinos caminadores o pepinos nadadores. Y una característica de este género es principalmente esa vela de 4 tentáculos que tiene sobre su cabeza.
Hizo popó. Y dio susto.
Ah, yeah, it's a polygon. Yeah, that is a.
Wow.
Mike is. Yeah, this is a polygon, polynoid polychid.
Thank you.
That animal is a commensal of that because that polychid feeding of the different, I don't know, the poop of the poop of that urchin or
Yeah.
that type of feeding of this urchin. Oh, urchin, sorry, sea cucumber. Okay, Mike? Polynoid. Now we're serving a swimming sea cucumber, genus Amperima. Yeah. Okay, Mike. Time to go in. Okay. It's possible to when we find one hill, sand hill, try to move to see that what is the animal that is hidden in the sand.
You can definitely do some digging.
Yeah.
Who has to do a push core?
Ah no, which one no?
No.
Do you remember the push core party?
Yeah.
1 hour and one push core party.
Yeah. One.
And now we're serving Aspidiodema urchin. That animal is very curious because his spine are very long and his body is very tiny. Una de las características, como señalaba, que estamos viendo hace poco era un erizo del género Aspidiodema. Cosa curiosa de este organismo principalmente es que tiene una... tiene unas espinas bastante largas y su cuerpo es bastante pequeño en comparación al largo de las espinas, las cuales pueden ser entre 2 a 3 veces el largo del cuerpo. Yeah.
Oh, you want to take these lumps?
No, try to move with the grab, with the arm. Try to move. Try to.
One of these mounds.
Ah, no, son montañitas de... Están enterrados, debe ser sibuncurios, porque esos que están ahí son desechos, son pellets. Ok, thank you. Mike.
You know what to do?
No, no.
No?
El estéreo. Ya, una vez que le diera
Sí,
la
está-
pantalla, ¿me puedes mover un poquitito para ver lo que lleva?
Okay. Give me the... Ah, no, that is...
I'll just take a good one.
Yeah. Now we are serving a sea cucumber, genus Ventodites. Maybe the species is a Ventodites typica. Yeah, that hill looks interesting. Como señalaba, el pepino que se vio hace un par de minutos es un Ventodites, posiblemente especie de Ventodites típica, especie bastante cosmopolita.
So you don't have to just dig. Just
Mhm.
dig with the tips of the fingers. You really get this on the other thing than to sound it out. That's how we'll talk about it. Maybe the beach you're doing this.
Yeah.
He wants me to poke around just with the tips. Who's in there?
No, that's- The other thing is maybe it's a hill of poop.
Hello, poop.
Yeah. Okay. Yeah, I think maybe it's a hill of poop.
I'm gonna carry on.
Yeah.
Yeah.
Bueno, recordando los highlights de la presente campaña, hemos visto distintos tipos de organismos de los que hemos visto en las campañas del presente verano. De entre ellos destaca la cantidad de cefalópodos de los cuales hemos observado durante la presente campaña. Durante las primeras inmersiones, como señalaba, observamos un pulpo Casper. Posteriormente a ello, observamos durante otras campañas, otras inmersiones, algunos pulpos dumbos del género Grimpoteutis. También observamos un Cirroteutis durante las primeras 2 inmersiones. Y un par de inmersiones atrás, no recuerdo si es la 695, 696, pudimos observar un registro de un calamar látigo, posiblemente del género Promacoteutis. Registro bastante extraño para la zona y es la primera vez que se observa el organismo vivo. Otro highlight de cefalópodo que observamos durante la presente campaña fue el avistamiento de un helicocangria que fue también colectado en la presente campaña, y lo cual nos va a permitir llenar ciertos vacíos de identidad o ciertos vacíos que existen en la biología y en la ecología de estos organismos. ¿Este pulmón se colectó?
Sí.
Ya.
No, por esta moto parecía.
Ok, Mike. Marisa, need you.
Mike,
So
remember,
there's the man.
remember.
Son is the man? Okay.
Remember
Okay.
this guy is gonna escape. is he?
Yeah,
Okay.
that guy can escape.
This guy can escape. Just saying. Do you need to get a bit closer? Maybe.
Remember.
Yeah.
And now we're serving the sipen. That sipen is Protoptilum. The was most curiosity of that sipen is that sipen can escape.
These are fun, so
Yeah. Como señala una de las cosas curiosas de estas, ¿cómo se llaman? Plumas de mar del género Protoptilum, es que a diferencia de otras plumas, esta puede retraerse y escapar.
Right, so you just have to get it lined up, look right in the middle, don't touch it at all, and when you're right down, like just in the seabed a little bit, just go. Like, full move.
No pressure, Tommy.
So nice first escape in the seabed.
Oh.
So you got more information than I have, Ken. Look up the first one he just went.
See, remember, Mike.
Yeah.
Yeah.
Exactly.
Explica cómo arranca.
In the moment we trying to collect that sipen, but to collect we need to be careful because that sipen can escape. Yeah, they can escape. Look at this. When you touch the sipen, this sipen is buried into the sediment, and are very quick and very fast to bury it into the sediment. Oh,
Ooh.
Got it.
No, no.
yeah, Sonny is the man.
¿Vas a hacer close-up?
Okay,
¿Sí?
always we need a glamour shot. Yeah, this is a coral or soft coral. This is a sipen, Penatuloidea coral. And this is a octocoral. It's very close to the Gorgonian or Pribnoid, Plexaurid.
Hey guys, do you want me to continue to this waypoint or you want me to stop?
The genus of that sipen is
Yeah,
Protoptilum.
You can continue on, Tony. Cheers.
Okay, cheers.
But with that picture is okay.
Yeah.
Yeah. But I need a picture of the peduncle or the base of that because this is a main characteristic of that. Yeah. Yeah, which queer?
El 16 está abierto.
Yeah, 16 is open.
Semi-open.
Ah, estaba abierto y entero.
That's alright, I'll open it together. Bring that camera in. That's it. Always too much to get in. I feel like we went all the way. We went- Chocolate in any or pork chocolate. Pork chocolate?
Thank you guys. Marisa will be happy because
new one.
that's it. That even looks new. A new one. I don't know. It's a new species. Maybe it's a new report of this part of the Pacific, or new potential new species. Que unos minutos atrás comentábamos que por qué estas plumas de mar pueden escapar. Porque a diferencia, por ejemplo, de otros grupos que hemos observado durante la campaña, Balticina, la Antoptilum, Foniculina creo que también hemos observado, estos tienen un axis o tienen una estructura de soporte que es bastante rígida, la cual no solo le permite retraer los pólipos lateralmente. No así Protoptilum, que Protoptilum tiene un pedúnculo el cual es bastante holgado y además carece de esta estructura dura de sostén que tienen otras plumas de mar, lo cual le permite enterrarse. Y por tener este tipo de habilidad pueden moverse ciertos centímetros y no estar en un lugar fijo, como a diferencia de las otras plumas que están enterradas y ahí van a quedar. Esa es una de las curiosidades con el género Protoptilum. Entonces, Una de las formas de colectar este tipo de organismos es principalmente tomarlas de la base para que no puedan enterrarse. Gracias, Mari. Ya vi el... Gracias. And now we are serving another CPIC. And also Umbellula cipens. Yeah, it's a cute CPIC. That is Umbellula. That's the other type of cipen. Okay, thank you, Mike. Are we in the... Oh no, almost in the waypoint number 3. Nos encontramos viendo un ambiente o una configuración de paisaje bastante homogénea, regular y continua, principalmente compuestos de sedimentos biogénicos, los cuales están entre una mezcla entre sedimentos limosos y arena gruesa, con algunos montículos o bioperturbación, los cuales pueden ser principalmente por algunos organismos enterradores tales como sipuncúlidos o algunas estrellas, principalmente del orden Paxilocida, que son estas estrellas que suelen enterrarse. And now we are serving 2 Hypnops fish. And the other one is there. Hypnops. IPNOPS. Now we are serving a Inopid fish, Hypnops genus.
You want it?
No, we have a one. Thank you, guys.
C-pen.
Only a quick zoom of that seven.
Oh, yes. Hindi lang sa kawan.
Yeah, we collect that seven before. Another Protoptilon. It's very different to the previous Protoptilon collected before. Another Protoptilum. That is curious. I'm looking for a Hypnos, a quick search in fish base. No, it's a hermit crab. An etymology of Hypnos means from the Greek Hypnos is asleep, plus ops, that is appearance. That is sleepy fishing.
Sleepy fish.
Yeah. That is the common name of the Hypnos, sleepy fish. Como señalaba, estuve haciendo una búsqueda rápida sobre el género Hypnos y el nombre común de este pez que pertenece a la familia de los peces trípode, que proviene del griego hypnos, que significa dormir, y ops, que significa apariencia. Entonces su nombre común sería pez dormilón, y ese sería el nombre común que tiene este organismo, Hypnos. Okay, thank you. Now we are serving a polychelid shrimp. A different type of uroids and Cicocumbers and Perima. Now we are serving another sea cucumber, but it's very different than the sea-pig. The common name of that, the genus of that sea cucumber is Aventodites. And in this moment, we are serving one anemone. That anemone maybe is looks like a seriantarid anemone, tube anemone. Observamos un pepino del género Ventodites y una anémona tubo. Características de este erizo, de este pepino principalmente, que su cuerpo es transparente y presenta una vela alrededor de lo que es el margen del cuerpo, permitiéndole esta vela también, al igual que por ejemplo Amperima, que tiene una vela cefálica. Estos pueden también utilizar esta vela para poder moverse y flotar en la parte de la columna y permitir ese nado o deslizamiento por larga distancia flotando. Género Ventodites, posiblemente Ventodites típica. And now we're serving a different type of hydrid colony. Maybe is Leptotecata or something like that. I need a quick close up. Yep. That thing in the stalk is Leptotecata hydrid. Okay, thanks. No problem.
Fast there. Updated waypoint glitch.
Yeah.
Señalado un fondo bastante regular y continuo de sedimentos de arena gruesa, algunas partes de sedimento limoso, cubierto principalmente por pepinos de mar, algunos ericios del género Aspidoadema. Y aunque este lugar parezca que no tuviera ningún organismo, se ve como inhóspito, la mayoría de los organismos de los cuales se encuentran en estas zonas principalmente tienen hábitos infaunales. Eso quiere decir que son hábitos principalmente que viven dentro del sedimento enterrado. A diferencia, por ejemplo, de las estrategias que tienen los organismos que habitan en zonas rocosas, que por lo general suelen habitar sobre la roca, entre grietas, fallas y fracturas de esta misma. En un ambiente arenoso, la fauna tiene que adaptarse según las condiciones de las cuales se presentan. Como señalaba anteriormente, la mayor parte de la fauna que habita este tipo de ambientes arenosos tienen un tipo de vida o una adaptación más infaunal. What is that fish?
¿Qué pez es?
Batipteroi. And now we are serving a tripod fish, Batipteroi, maybe Batipteroia tricolor. Okay, no problem. Yeah, no problem. Keep moving. This triple fish is the same family of the Hypnoph family. Sorry, the Hypnoph genus. The difference between the Hypnophs and the triple fish is that the Batipteroi has long fins, pelvic fins, and dorsal fins are so long. And the Hypnoph genus, the main characteristic of that fish is the big eyes, the big yellow eyes. Entonces en este momento estamos viendo un pez trípode del género Batipteroi, posiblemente Batipteroia tricolor. Es bastante cercano, de hecho es de la misma familia que el pez que vimos anteriormente, Hypnops. Una de las características principales que diferencian a ambos géneros es la presencia de aletas, y que las aletas pélvicas y aletas dorsales tienen proyecciones bastante alargadas. Asimismo, el género Hypnos tiene grandes ojos que son de color amarillo, careciendo de estas prolongaciones que tiene el género Batipteroi, las cuales también dentro del mismo género Batipteroi pueden ir variando según el largo y las divisiones que tengan estas proyecciones. Thank you, Mike.
Sandy sea beds and all. You have to fly
Yeah.
so close.
I would like to found Octagnemus. That is my favorite deep sea animal, the carnival tunicate.
Parece humano que tiene un otro. I don't think
Yeah. Can you zoom in on that?
Que andaban por tuviera por aquí.
That's a problem.
Yeah. There's only a quick zoom of that. Okay, thanks. That is perfect.
Porque no aquí de la tequila de oro.
puede ser de, no de pepino, de hecho podría ser de algún torcuaratari, de algún enteroneuta. Que dejan esos trails, suelen tener esos trails redondeados. Mm, the worm shape that we saw before, maybe it's a species of torcuaratari worms, enterneuta, or acorn worm, sorry, maybe it's a species of acorn worm. Or maybe civic. Aspidodema again. Another amperima. ¿Colectaron amperima?
Hoy día,
Mm.
entonces pusieron a
Okay, can we collect that CPIC?
de chiquitito,
Yeah.
sí,
You don't lose all
y
the
una
news.
succión.
No, no, Susan lo va. Se pueden tomar.
Ah, vale. Sí, dale, si no te
We can
nada.
grab it. Ah,
Can't use the socket for this one. Too big, maybe.
¿Cuánto mide? 12 centímetros.
You use no best for you. The dust. We'll get the next one. Yeah. Just kind of mess it out too much in
yeah, no problem.
the sand.
We can find another.
Sí. Sí, vale.
Next one we'll just snatch up. You know, you want to know what
Okay, now we are serving a Bathyptylos fish. That is a Inopide fish. It's the same family of the Bathyptero. It's a Hypnoph fish. But the difference between, for example, Hypnophs and this one is that Hypnoph has a big yellow eyes, and this fish don't have eyes. That is very curious.
¿Ya cómo se llama?
We'll wait and see if we can even catch this guy. He's fast. We gotta be more catch him. He's pretty fast so far.
Yeah, no problem, but with the good pictures, okay? But during the Salas y Gomez campaign, we saw one of these. But the difference between this is the other one is have a other color. It's a different. That is a most darkness than the other.
Right in the circle.
Ah, espérate, te doy el nombre del tiro.
Right in the circle. They move the vessel off. There. You took in some where? Yeah. Yeah. Now you got people in Nazca there. Updated waypoint bridge.
What? Ah, that is octanemid carnivore tunicate, but it's the first time that we see at this time of tunicate in that part of the Pacific. Maybe it's the genus Situla. This is a carnivore tunicate of the family Octagnemidae, but it's very similar to, for example, Megalodicopia or Dicopia. That animal is very weird, and this is the first time that we see this type of or carnivore tunicate in that part of the Pacific. Ese parece que es Claudia Situla. Situla.
¿Es el que tú querías? ¿El que
Eh.
te faltaba?
Sí, ese es el que falta. During the Atacama Trench campaign, they saw this type of tunicate very close to the continental shelf of Chile. Yeah, that is very weird.
Ya, conéctalo.
Have
El que te
the-
faltaba, conéctalo al tiro.
So when you were what is it now?
Yeah, that is very fragile.
¿Me puedes escribir el
Situla con S, S-I-T-U-L-A. Situla. Situla lanosa, posiblemente. Yeah. Yeah, correct.
What do you want? Trying to put it in a bio box?
And biobox
How do you take it?
for this part, because if you take off the base, you have this boom. That whiplash.
So,
So
with
just
the
grab it in the
The,
middle.
I think that, yeah, the middle of the stone.
You got each of it in?
Mhm.
Yeah.
Look at this toy. I read
Yeah.
one note of that,
Oh,
and now I have this one. That is the same in that part of the Pacific.
okay.
It's cool. And now I have 5 species of the Nemidae family.
Crazy. Crazy.
SITULA. Huh?
Is that right?
No, not this megalodicopia. Looks like a megalodicopia, but the difference between megalodicopia is the shape of the mouth of that Octagnemide, because the megalodicopia has the mouth similar to the Pac-Man shape, and this one have a mouth very similar to a spoon. But I'm sure that is Octagnemide tunicate. But this is the first time that we have this type of Octagnemide in this part of the Pacific, because we saw four genus for this area. The first one the first one is like a flower is Octagnemus, and the second one we saw a megalodicopia, and after that we saw a dicopia also.
Happy like that?
Yeah, happy with that. Yeah, because I have a picture of the peduncle and
Okay, so you just want the top there.
yeah. It's okay. I don't need a.
Yeah.
Okay, and after my.
Yeah. Hold on a
Okay.
second. Maybe you have to come up a little bit.
Bueno, como decía, este es un género de tunicados carnívoros entre los que hemos observado durante esta campaña. Hemos observado uno con forma de flor, que es del género Octagnemus, uno con forma de pedunculae con forma de Pac-Man, conocido como megalodicopia, uno sin pedúnculos más corto, conocido como BV3V, is okay.
Maybe.
And after that, can we collect BV3V?
V3?
Yeah. And after that, can we collect the peduncle of that and put it in the same curve?
Yeah.
This type of animal is more related with the ascidians.
Really wanting the bottom there.
Yeah, maybe
Because-
change the box and BB2B. This is much bigger than the BB3B.
I think the problem is it's gonna float.
Yeah.
so that I can come up, and then I don't know if we're going to be able to watch the edge of the box, man.
Yeah, but we have patty pattes in the BB2A. I think maybe in BB2B it's okay.
Well, that's what I mean. If I come up, we're maybe not going to find the other half of it. You know what I mean? I'm thinking about coming up so that it goes into the box, so it's trying to float just now.
I think maybe it's float if you.
So if I come up, it'll go down, but then I might not get the
Okay,
other half. So it's up
we
to
try.
you.
No, what could we not just put a fix where the RV is right now? So like it's
Station keep, but
No, on the nav. We can,
I just
Make
think
a
where that
fix.
station is a fix.
oh, okay.
So
Alright, it's not coming up easy.
it's just what is it? It's just floating. Watching light.
Open it. Come on.
Oh, be careful.
You're on the thing.
Okay, is it?
Close.
Yeah. Oh no.
Oh,
Okay.
It's right there, man.
No, I think maybe put it in the bio box 2B. Try to collect off this part because that is a main character.
Which one?
For that part because this part is the main character to get to identify the genus of that animal.
Nice and easy. Right, try and put it in the same box. I don't think it's the size of the box that's the problem, it's how floaty it is.
sí, porque flota demasiado, dice.
Sí, pero que decía que lo cambiáramos al BB2B.
Es que él dice que no es la caja, lo que te dicen que no es la caja el problema, que flota demasiado el animal.
Que si se mete dobla, baja y cae, se quedaría ahí, pero como la caja es muy pequeña no se puede cerrar.
Ahí dice eso. No sé.
Raise up a little bit.
You're clear, you're on the side of the box.
Come on, then come over here.
But I'll try and close the lid quick if you manage to get out. It's
Okay.
thrusting up all the time. Just need to come off the side of the box. No.
Oh,
You know, Michael, you did your best. Quiet.
mm-hmm.
Squeeze there.
What? It's there?
close this box when you get to the top.
Yeah. See it?
Give me a look for it.
That is.
Guys.
Oh man.
Now it's into the rock box maybe. Me lo puedo darle todos los caracteres.
Estoy tagueando yo también.
Gracias.
Keep my guy just come out.
It's gone? Oh,
Yeah.
is there? It's gone again.
So forward.
Stay left a little.
Keeps getting caught through still options.
Uy, uy. Salió.
Why does he manage to like go fast sideways whenever I'm coming up? It's right at that moment where he's getting caught with the
Yeah.
direction of the twisters. Well, I move the vessel off a little bit, mate, because I'm like so close to the 58 here.
Yeah, just move it.
Is that good?
Yeah. Bridge updated waypoint.
Close, close,
Close,
close,
close.
close, close, close. Come on, please. Oh my god. Oh, no, yes.
Oh, got it.
Yeah.
Amazing, Mike.
I'm gonna go to the options, yeah, okay, keep going.
Oh, wow.
Okay, thank you guys.
Roger, never mind bridge, we captured the,
Baby
we're
3D.
trying to capture that.
Thank you guys.
Yeah. Do not open.
Okay, and after that, move to the.
Then we'll go down and get the other bit.
No, no.
What do we call that again?
What nodes?
Tunicate.
Yeah.
Don't open.
Okay.
Is firebox 3 full?
Yeah.
No. Just has on the M3A. B and C is. Alright, tuna open.
Okay, thank you guys.
What's the chance of us being still over the other bit?
Okay, Maxi, la sample está hecho.
Okay, I don't know, man. We tried to keep moving.
Follow your track.
And with that part of the tourniquet, I'm okay because in that part of the tourniquet I have the main characters to get a
Cool.
genetic analysis and morphology analysis.
Store the arm and stuff, I understand. Yeah. Tricky.
Yeah.
It's hard to hear.
Yeah, that animal looks most easy to collect, but was a old challenge to.
Too floating.
Yeah.
Me llega en el chat que no está completo.
Sí, sí vi, pero. I'd like to remind you that the old specimen or the old sample collected during this campaign will be a legacy for future scientists and researchers. And the samples will storage in the Sala de Colecciones Biológicas in Chile and the Natural History Museum of Chile. To protect this area, it's very important to know or understand the process, the characteristics of the habitat, and also the most important thing here is try to understand the biology of different type of animal that live in that part of the Pacific. That is the reason that we collect this type of animal in this part of the Pacific.
Ellos mismos te.
Bueno, como señalaba, que la colección del material, el cual estamos, o animales que se colectan en la presente campaña y en las campañas que se hicieron durante el verano, no es que se colecte por colectar. Gran parte de la información que aporta los distintos animales que se colectan, estos son preservados y mantenidos en una colección la cual se encuentra en Chile, que es la Sala de Colecciones Biológicas de la Universidad Católica del Norte y el Museo de Historia Natural. Con el fin de entender la biología y la ecología de esta zona, es importante tener información acerca de los animales que viven en esta área. Es por ende que los organismos que se colectan son debidamente procesados y debidamente preservados para que sean un legado para futuros científicos y esta zona sea protegida de la mejor manera.
¿Vino?
No, only a quick zoom of that.
¿Qué?
Solo una buena foto y chao.
Sí, pero ¿cómo?
Amperima.
Amperima.
Is it the same guy that you want?
Yeah, the same guys. Yeah. Yeah, okay.
You want to collect? No?
No.
This guy.
Oh,
That's what I meant. That's what I thought.
Again, also, I would like to remind you that the main goal of this campaign is to explore the seamounts of the Nazca region international waters and aiming to explore remote and uncharted places to provide information to protect this fragile and remote ecosystem. Sinafogranquido.
Sinafogranquido.
Sí.
Con C, con S.
Sí, con eso te lo escribo.
Wow.
Va. Me gustaría recalcar y recordar nuevamente que el principal objetivo de esta campaña es explorar los montes submarinos de la cordillera de Nazca en aguas internacionales, con el fin de explorar y promover nuevas políticas que las cuales puedan proteger esta prístina zona.
Yeah.
What happened? Ah, do you need a rock? Okay. Okay. Yeah. Now we are serving a Lobata flow. See you later, Tomer. Thank you for all.
Have a nice
If you need something,
Ciao.
just write me in the chat.
Ciao.
Yeah.
Ciao.
Ciao. Ciao. Looks like an Italian.
Yeah, Luca.
Luca. Ah. Oh. 1 minute for Luca. Ciao. Yeah. Kojo Suminov. That is the maximum sum or?
No, no.
Ah, sorry.
¿Es una pluma?
No lo sé, tengo que ver.
Qué bonita.
Sí.
Es distinta. Esa sí que es distinta.
Sí.
Well, nice bit where zoom for this.
Okay, and finally, I would like to remind you that we are in international waters, and therefore the fauna of this area has no protection. By collecting samples, we will have a more evidence and arguments to protect these zones. Como señalaba, y finalmente para recordar que nos encontramos en aguas internacionales y por ende la fauna de esta zona no cuenta con alguna protección. Es por ello que colectando este tipo de organismos nosotros podemos tener más evidencias y argumentos para poder proteger esta prístina zona. Wow, that looks weird.
Sí, ¿no? Para Marisa.
No, no es una pluma, no tiene pinta de pluma.
¿Qué es?
No sé.
Do you want me to
Yeah.
land so we can get a better look?
What is the side of that coral?
Lying close to the wall here. Lasers on. On the land. It's getting tricky.
Now we are serving a one coral. And the rocks looks like a pillow lava or lava pillows. Sí, sí.
That's a Mark II, that's terrible. A little light on it.
Okay, guys, can we collect that?
That's too dusty. That's just hands about, is it? Okay. You just want it in a coral cover?
Yeah, 18 is okay.
I think we put some 16. Yeah, 16 last time, 17. Overhand's quite humble.
Qué raro ese coral.
Sí, está.
Porque se levantó bien.
Get a good shot here. It's too sandy.
Sí,
Mhm.
súper sandy. Is it the current or
The lacquer current, I think, and
Oh.
just the sandy, fine seabed, and then as soon as it stirs up, then it just hangs. where it
Right.
is. As soon as we're having to thrust about anywhere near it, it just
Right.
stays.
Ya está bien nublado, así que.
Oh, weird.
Yeah. Oh, that color is so weird. I don't know what is the name of that color. Let's see.
Wow, cool.
Yeah.
Oh, que lindo, Sebastián.
Yeah, I'm super happy with that. But I'm super sad because I don't know what is that.
Estos are cool.
Gracias. Yeah, that thing looks like, I don't know, macroprypnoa or something like that. Okay. Después lo vemos en el... Okay. Coral quiver number 17. Okay, thanks. Thank you, guys. Sí, batipates.
Yay!
Yeah, the coral was the coral collected before. Maybe it's a pripnoid, maybe it's a macropipnoa, but I'm not sure. I'm super sad for that.
Ese es tu ego. Calm your ego. Man.
No.
Jesus. You're human.
Yeah, looks like more a black coral or, but I have a doubt because the polyps of the tentacles of that coral have a pinnus, and that is a main character of the octacorals. Okay. Now we are serving now Ormati the anemone.
¿Cómo se llama? ¿Amatia?
Ormatida.
Ah, Ormati.
Ormati de. Está aquí, bueno.
Sí, ya sé.
Ah, ya. The main characters of that anemone is that has a cuticle cover the body. Resinged star. Crinoid.
Mhm.
Okay. Resinged star and crinoid. Another large crinoid. Another crinoid. Antonidea superfamily. Rising star. Ofecante de breeder star.
Wow. ¿Eso qué es una esponja o un tallo?
Sí, un tallo de esponja.
¿Está muerta?
Sí. Increíble lo resistente que son. Are we in the waypoint number 5 or almost in the half?
Yeah, almost there.
another Risinger star. Sorry, another of your retro star. And no, that's looks like a tiene sifona, está apagado, oh, está rara ese poco, pero por la raíz puede ser una ferronemátide. Wow, how many ophiuroids and crinoids, muchos ophiura, posiblemente de la familia Ophiopintaca, Ophiocantide, lo siento, con crinoidio. ¿Ah?
No se me están guardando
Los ophiuros hemos tenido, pero
no,
que...
no. Pensé que no me estaban funcionando a mí en su lectura.
a veces se traba, como que
Sí,
desmoran.
no,
Pero sí, yo estaba viendo tus comentarios. Datipates. Ophiuros. Another Sipen. Protoptilum.
Same one.
Yeah, the same one.
Wow, mucho estudio.
No. Es Tauropates. Sí. No, eso que está ahí no es un Sipen, no.
Interested or
No, no.
no?
Chrysogorgia. Sí. Bueno, dilo ahora.
No, porque yo quería hacer. O sea, yo sabía que era transmucho. Lindo el color.
A few minutes ago, we saw a one carnivore sponge. thought about this. Esa que está ahí es una esponja larguísima. Así.
Mira, mira, ese,
¿Cuál? Ah, los Furious.
muy arriba. O ya pasamos ese, no, todavía no. Ese palo. La esponja muerta ya habíamos visto ese, o es la misma o no? No.
No, no. Que acá hay muchos tallos así que están de estas esponjas que podrían ser algunos tallos de bolosoma. Bueno, seguimos aquí en nuestro recorrido, cambiamos de un hábitat arenoso, regular y continuo a un hábitat con lecho rocoso, con algunas estructuras o depósitos sedimentarios, observando gran parte de fauna incrustantes, tales como algunas esponjas y algunos ofiurios. Oh, está...
Okay.
Yeah, thank you. Wow. Riddle Star
Oh,
Paradise.
No puedes walk.
Oh,
Okay. Mike, can we collect this fury
Yeah, which one?
and that
One or
that
two?
one? Anyone? Yeah, maybe two in the Korakura, 18.
Sure, so I can run John. Not fibers. Hmm? Whatever one do you think is worthy?
And a few minutes ago, we saw
Are we gonna speak corals and stuff?
one Ascidian tunicate and the family Puridae, Culeus, Culeolus tunicate.
Quiver also.
Don't know if you people get in a quiver, but we could open one up.
Oh, shall I build it?
Era
Open and
el
see, but I'll probably be my box.
Pooch. Pooch.
¿ese es el Furio?
no, esto es esto. Es un tunicado.
Ah.
Yeah, a few minutes ago we saw Culeolus tunicate, Culeolus. ¿Qué te salió?
Si quieren poner a... Javier dice que como no tenemos a la cera, Javier propone que toque algo de heavy metal para la despedida.
Go reach them now.
Yeah,
Getting
can
arrested.
can we slurp that brewster? I think
Yeah, you probably could.
yeah. Because are super bitter.
Yeah, we can give it a try with a slurp. They live up to their name, to the little stars. That's true.
It's of your canti the family of that of your this one. This type of ferret.
They get called little stars.
Yeah.
Thank you, Maxine.
Okay. Si, oh, si ves algo así, mejor intentar colectar porque este era muy grande para la.
¿Es una esponja?
No, es una asidia.
Maybe como un tunique.
Un tunique, sí. Este es el género. Mm.
Ok. Sí, podemos encontrar una que es más pequeña.
Sí, y eso con eso ya es la primera vez que se ve algo así en esta zona. Vimos un par atrás, pero eran gigantes. Y no se puede abrir este body box porque colectamos uno de estos y se abre y se va flotando. Entonces, mejor tenerlo acá y va justo con el aspiroderma que colecta. Conectamos hace poco un protoctilo, ya el protoctilo ya no más, están todos listos. Un coral indeterminado, entonces no sé, puede ser un plasmoide o un bambú, o sea, tiene una forma bien rara. Y ahora vamos a conectar estos fosfurios que están por están cubriendo toda la roca. De hecho, desde que empezamos a subir, están de esa manera, en la roca. Entonces, ya eso ya, claro, empezamos a conectar. Sí, y 19 todavía no estamos casi llegar al punto 5 y recuperé 10 minutos, así que estamos atrasados por 20.
Ok, no, está bien. Sí, ¿cómo dejaste esto? Ya, está bien. Gracias. Tengo el vaso. No, está bien. Oh, that's loud.
No sé,
Stubborn.
yeah.
Yeah, se fue.
Which section is it?
Number one.
Uno. professor at the University of Texas, Rio Grande Valley, and I'll be here with you for the next couple hours. We're just here collecting some more chlorites that are all over this rock here. Thank you. Yeah. Let's just try and grab another one, I guess.
Erin, the suction is ready anyway.
Okay, perfect, thanks. Yeah, with the suction.
Dale, Maxi, vaya a alcanzarlo.
I'm gonna log the sample here.
Empezaron a salir turicados, primera vez que vemos turicados en el monte.
Mira.
Así ya no lo hemos visto hace mucho.
Erin, they're saying that your mic seems far away. That you're not
Oh, okay, yeah, I'll fix it in a minute. Thank you. Appreciate it.
They break so easily.
Yeah, they do, but we can get the discs, that'll be good. Yeah, I'll fix my mic as soon as I finish logging the sample. Alright, great. Yeah, that's probably enough. I think we have, is that 3 partial individuals? 2 now? Okay. Yeah. Yeah. It's like, we'll use the discs, I guess. It's fine. I think the discs have good information. Reposition this so my mic is better. There we go. That should be better. Okay. Alright, thank you very much. We just arrived at waypoint 5. Alright, so we're doing a shift change right now. I've just come in from Maxi. And there's a crinoid down there.
Yeah.
Like Tyler and Ben are coming in for Mike and Sunny. So just passing on information from pilot to pilot before we get going again. If you still can barely hear me, that I don't know. My mic is close now.
updated waypoint, Reg.
Copy that.
Okay, great. Good to know the mic is in a better position for you all. Tyler, we'll just keep heading along the transects with lasers on. Thank you. What is this? There's like snacking and stuff. Oh, okay. Awesome. Thank you. I'll store that for chocolatey, chewy. I'll keep that for a little bit. Thanks. Alright, so yeah, we are on our second dive with the Seamount. It is our last dive of the expedition. And we are at a deeper location than our first dive, which was on a ridge heading up to the summit. This one we started out around just over 3,000 meters depth, and we're going to end up around 2,000 meters depth. So we're a little behind schedule. So we can move along slightly faster, so we can recuperate some time while we're over sediment. So we are on a Seamounts on the Salazar Gomez Ridge near the junction point with the Nazca Ridge. We are on the northern side of the Seamount, following the ridge southward towards, so it's a little off the main part of the summit. We're working our way up a series of small, kind of knoll-like features along this ridge until we get to the main part of the ridge around 2,100 meters depth. Yeah, shrimp. Always shrimp around. There's some nice tracks there that is like really tight S-curves. seeing some crinoids here and there. Anemone down there. And it's a lot sandier here than I was expecting for the ridge structure that we were seeing on the mapping data. I thought we'd have a bit more exposed rock. But we are pretty deep, so maybe it's just been raining down. Oh, looks like we have a snailfish. Or no, maybe actually.
What is that? Looks like sea cucumber.
No, I thought it was a snailfish at first, but now I'm not sure. Let me see. It might be a flatworm.
Oh.
Yeah. We can just get a nice photo of it.
Yeah. Hi.
Zoom closer.
Oh no.
He wants to. It's really trying to see right on the left. It's right there.
Hey Rich, can you check what the mic volume is? Because I'm speaking the same volume I always speak and...
Turn it up.
Okay, thanks. So, I don't know if like a whole bunch of people turn down their volume at home or the mic, what's going on. Thank you.
Pelagic, never.
Yeah. Awesome. It had the coloration at first of a snailfish, so I was like, oh, but usually they're all curled over, so. Nice. Nice size sea cucumber over there. Still seeing some of these sea pens that we collected early on the dive. Another. Yeah, we can zoom in on these two sea cucumber. It's a sea cucumber.
Nice color.
It is a nice color, yeah. Then the sea cucumber that we got a nice close-up earlier today of, a bright yellowish one. Yellowish green.
Oh, another one.
Yeah. Look at the, huh. Look at, like there's a worm moving along in the background there. Small one. Awesome, thanks.
What is that?
I'm not sure from here. Yeah, I think it's just another one. So yeah, we can.
Fossil?
Yeah, I think so.
Oh.
We have quite the collection already. We're good. Yeah, another sea cucumber. Let's see, waypoints. Alright, so we're nearing waypoint 6. So make us about a quarter of the way for our waypoints for today's dive. Can we zoom in on, I think this is a glass sponge, but can we zoom in on it? Just for the lasers. Yeah. Probably very possibly the species that's leaving behind a lot of these stalks that
Maxie
animals are
told me
settling.
that- No, this is a sponge.
Yeah,
right?
this is a sponge. Yeah,
He
it's not.
said he's looking for this.
Yeah.
So, it's not a tunicate.
No, it's not a tunicate. No, it's just a glass sponge. Yeah, we're good. Thank you. We're just looking. There's a tunicate that Maxie wants to see wants us to collect if we see another one that's smaller than the last one they saw. I think maybe it would normally be on rock though, but I could be wrong. He just showed me a quick picture. If you see cucumbers, we have at least 3 or 4 different species that we've seen throughout this dive. Glass sponge. That's another sea cucumber. That might be the fourth or fifth different species. There's what we refer to as an empanada crab. It's a crab that wears some anemones on its back.
Oh,
These ones.
first time that I see one. Oh, that's so weird.
I know, they're so interesting.
Yeah. Wow.
Yeah, it's.
Has like fire on his antenna.
It's cool.
Very cool.
Yeah, we usually see him walking away. Just kind of hanging out here. Lovely. Thank you. These are actually zoanthids on its back. Not anemones, sorry. I misspoke earlier, I think. There are crabs that wear anemones on their back too, but it's a hermit crab. Brittle star, crinoid. Another area that's covered with these ophiurids we tried to collect earlier. Looks like a little glass sponge there. I always find it so fascinating how one moment you see one ecosystem, one group of animals, one community, and then just moments later you see a completely different one. I mean, huge abundance of these ophiurids like we're seeing now.
Yeah, so many.
Yeah, I know, so many of them. And it's in,
Crinoids as well.
some crinoids mixed in occasionally. Broken sponge stalks. It's funny. They just like there's just on the sides of some of the rocks, but not on all of them. It's like they're waiting to jump in the pool.
Yeah.
Sitting on a ledge.
So strange.
Yeah. I see a cucumber there. Some small coral whips we're seeing as well. I think that's a good sized glass sponge up ahead. Nice. Really beautiful one. These look to me like they're broken up lava sheets.
Nice.
Yeah.
Amazing.
It's all along the edges of these rocks, the ophiurids. It's oafs just hanging out. Occasionally some crinoids. Some of these stalk sponges. Still seeing quite a bit of sediment deposition. Another nice sponge down there on the side. We're seeing also occasionally these small whip corals. Not very big. See if I can find one in a good position for us to zoom on in a little bit. Yeah, can we take a look at this coral here? Let's see what species it could be. Like a large gooseneck barnacle on the stalk as well there with the oaks. Yeah, it does kind of. It's like, yeah, just left of the lasers there. This looks like a small bamboo. I think I'm seeing a node there. Let's see when it comes into focus. I don't know. Is this what they collected earlier? Do you know? Let me see if I can go back.
No, it's not.
It's not?
I'll send you a picture of the one that they collected.
Okay, thanks.
No, I'll see forward with that.
The one that Maxi doesn't know.
Yeah, can we go ahead and collect this coral? Thanks. I've been seeing it quite a bit. I think I might have seen a node there, but
This is the one that we collected earlier.
that's the one we collected earlier.
And it was really hard to see because it was.
It is interesting.
It is really interesting.
Back up a little bit. Just dial to the left. That's
Yeah, I thought I saw maybe there was a quiver open already that they'd opened. I'm not sure. I think 18 looks open.
Yeah, 18 looks wide open. Oh, that's Andy.
Yep.
Nice nose.
I think I'm finally starting to wake up. It took like such a hard nap after dinner.
Some hires here.
I don't know. Maybe this is the same.
Yes. I was gonna say.
Now that I'm seeing it, we can go ahead and collect it. Let's just collect a second one because we don't. Yeah, go ahead and collect it. I'm just
Yeah.
trying to figure out if it's one that was collected earlier.
I think it's the same one.
Yeah, but we can do. They're small colonies, so we can do with the second one. But it does look like it's some type of bamboo because I can see a node there.
Mm.
Oh, it's weird though. Yeah. So yeah, we just. Yeah. Normally we wouldn't collect two of the same species, at least that we think are the same species on a dive, but in this case I think we should have it. Yeah, let's go and get another glamour shot. They're small colonies. Fascinating. We have sclerates there.
Yeah, it's the same one because it's polyps are.
Mm-hmm.
Now it looks like the one that we collected.
Yeah. Yeah, it didn't look the same until we went to go collect it and it started tucking in. Let's pan down. See that's where I think I see a node right there between these two polyps. Yeah. Nice. Yeah, great, thank you. It's interesting, they kind of like fist up a little bit, the tentacles. Can really see like the symmetry really well.
It's coral cleaver 18, right?
Yep, 18.
Sample ready.
All right, thank you. I got the sample logged. Awesome, thank you so much. And then we can continue transecting along. Let's see what the, ooh. What did, Yeah, what's here on the left? Is that a weird?
Let's check it out.
Yeah.
Chrysogorgia?
Yeah, it kind of looks like it might be a Chrysogorgia. With the shadow, it was just.
So it's in line.
Yeah, just give it just a quick zoom. I think it's a Chrysogorgia.
Pretty
I
high.
can't believe I'm.
It's a pretty good sized colony. Yeah, here's the
Maybe otherwise.
gold skeleton. It might be a pseudo Chrysogorgia.
Oh, okay. Is that
Alright.
squatty inside?
Hmm.
Is that a squatty inside or?
I see an oaf.
Yeah, me too, but
and
there's
the
not.
other might be a squatty. It's kind of an awkward angle.
Okay, if you just stay there, and
Oh, I saw the squatty down there at the base, almost.
base.
Yeah, there's a squatty. It was moving, now it stopped, but it's almost at the base. Now it's moving its legs. See, there's the squatty.
Oh. Yeah, that's a squatty. I was seeing something higher up that I was
Yeah, but
looking
I also,
at.
yeah, me too. I know, I just saw it.
But yeah, I think it probably there's this purplish that may also be another squatty, and then higher up there's like a darker reddish
Yeah.
pink thing.
It is.
Yep. And then it's another squatty.
it's another
Yeah, I think it's like the one at the base, but I think there's something different further up. The bright red. Pan up a little further. Like above the oaf, yeah.
Yeah.
Like this right here.
Oh, so 3 squatties.
I think so, and I
Wow.
think this one's different than the others.
Yeah.
Yeah, cool,
Wow.
awesome. It's too big of a colony though to collect to get all of them. So, all right, onward we go.
Onward.
Thank you. Rocks right, left, and everywhere.
That's everywhere.
Oh, we're huh. Yeah.
Does, do you prefer to just cross it to the other side or you want to spend right here?
It's easier for us to cross the crowd.
Yeah, are we passing like a valley or something here? I'm sorry, I can't see the map. It's just a black thing right now, I think.
People like that. I'm just gonna bring this ship
No,
over
I think
closer.
it's okay. We can just mid-water for a moment here
That's-
until we get to the next. We're seeing a lot of the same right here, so.
I'm gonna bring this ship just up here closer to.
So some of these rapid descents like this are a little dangerous for the ROV, so we're just going to stay a little further off bottom and cross over to this next feature. Or at least till we get to an area where they can turn and head back up slope.
Ridge
So-
RV.
narrowly ridge.
Yeah, Eric, can we lateral to starboard about 20 meters?
Sure enough, go around to that new waypoint.
Roger that.
Alright, sounds good.
So while we're going downslope here, we're just gonna stay a little further off the bottom until we get to the point where we're going back upslope. This area that we're in has a lot of little knolls and different geomorphology here. That's a little complex, so there's just some locations where it's easier for the ROV to stay further off bottom, so it's in a better position to not get entangled. And so we're going to just do that for the safety of the ROV until we get to a point where we can work our way back upslope. It shouldn't be too long. It's kind of we do our best to set the waypoint lines along the track, but sometimes we need to hop from one feature to another. So we can make, in this case, we're making our way to a main ridge. Which of these levers lowers the seat? Oh, there we go. So much better. Sitting up too tall, making my have to bend my back. It's much more comfortable. Yeah, so it's looking to me like some pillow lava-like flows here. We're still seeing quite the rain of sediment, which usually means we tend to see fewer organisms.
That sponge.
Yeah, it's a nice little sponge. Can we actually look at that really quick? First I thought it was just a sponge stock, but I think there's something on it.
Yeah.
Looks like maybe it has some zoanthids on it. Yeah. Can we go ahead and take a piece of this and put it in a quiver?
Is
Thanks.
that like a dead sponge? with
It's a dead, it looks like a dead sponge stalk with some zoanthids.
zoanthids, right?
Yeah. So yeah, just a quiver-sized piece off the top.
Size piece. Hey Eric, can you come to unload stuff here?
What's that? Thank you.
Porch panel? Sure. How big of a piece do you want here? 3 inches? Just top.
Yeah, like 3 to 5 inches.
You can put that in the
Yeah, on the quiver.
cover.
Yeah, quiver.
Front row or side row? Side row.
Yeah. Yep. Like we've got a.
Is that sufficient?
Yeah, that's great. There's another, like a worm or something. I think we can skip the glamour shot just because I think I'm a little worried it's gonna break. Okay. Thank you. Coral quiver 2. Just gonna log the sample.
I'm doing the sample.
Okay. Yeah, I'm just. Thank you. I see it.
Done.
Perfect. Thanks. Yeah, I think the quivers are just under 10 centimeters. Diameter, right? They're like 3 and 8, like something like that, a little bit. And tall, they're probably what, like 15 centimeters ish. All right, thank you. Onward we go. Yeah. See what's at the top of this next feature. Rich is working on getting the map updated for us.
The bridge we're all done, so it's the marker. Unless I'm facing.
Alright, so we're just moving up slope, getting an updated map. We're kind of making a jog over towards the main ridge now. That's why we're not really going up slope, we're kind of going along slope. Hey Maxie. Yeah. Yep. Alright. We're just doing a shift change here. Max is coming in for Claudia. Yeah, it's a nice sponge. Yeah, it is a big bowl solo.
Okay, thank you.
Mm-hmm.
Huge. Oh my god, it's
Yeah, there's the other lasers. 10 centimeters, so 20, 30, 40, like 60, 70 centimeters across probably at the widest point. Yeah, you can go ahead and turn the lasers off if you want. Do a glamour shot. kind of looks like, because it's got the 5 lobes, it's kind of like a sea star. Reminds me of a sea star hanging on by just a couple feet.
What is the size of that?
What is the size?
Es como 60, 70 ancho. Yeah, it's pretty impressive. They're gonna do a glamour shot here. It's a little yellowish, so I'm wondering if it's not doing so well. Look at that small little sponge behind on the rock. Yeah, here I am. Can we actually go look at that really quick? I'm sorry, I know it's like in the opposite direction of where we want to go maybe, but it's got some bright little spot. I don't know. I think it's an anemone maybe. But it's got kind of these reflective or what? Okay. Okay. Corallimorphus. Corallimorphus anemone. Awesome. Thank you, Maxie. You can pop the lasers off, so there's a few seconds without the laser. Amazing detail. All right, great, thank you. Onward we go, thank you. Nice peach colored crinoid over there. Little urchin. Nope, maybe that's another crinoid. Yeah. Oaf down there, a whitish one. left. The Brazingid Sea Star that works, I think. Actually, no, maybe it's another crinoid, actually. Yeah. Looked in the sit cam like it was a Brazingid. It's a crinoid. Nice. Going up kind of a cliff-like structure here. It's gonna move up it carefully. See another one of these whip corals that we've been
How
seeing.
do you finish that?
Hmm? You wanna zoom on it?
Yeah.
Yeah.
No, we have had two of that, but I don't know what is the genus or family of that.
Yeah.
But only put some of that.
Yeah, let's do another zoom. I think in the segundo question me parece que tuvo un nudo.
No.
Like,
Maybe Vancouver?
like I think, yeah.
Yeah, maybe it's
But I only saw one possible node.
Yeah, maybe it's a Keratitoside S1 clay, or maybe, but not looks a primoid. Looks more than the, I don't know, bamboo coral.
Yeah.
Thanks.
Yeah, thank you. Yeah, I'm not seeing a node yet on this one, but on the second one we collected, it looked like it had a node. Just one though, so I don't know. We'll get a better idea when we have it up in the lab. Nice size crinoid there. Yeah, these small white whips are a coral. We're just, neither Maxine or I can quite place it for sure. So we're going to have a better look at the coral when we bring it up into the lab. Yeah, I think our off-bottom time is in 7 hours, about, depending on how shallow we make it. About 7 hours of on-bottom time left on this dive. But we'll be streaming the ascent, so the total time should be a little bit longer. Can we zoom in on this here? Thanks. Because I think in about 8.5 hours, for at least about 8.5 hours from now. This is a black coral of some sort. Yeah, if we can sit down, I think we're gonna collect this one. 10 centimeters. I think. This doesn't look like any black coral we've collected so far. We settled down on this dive at around 3,050 meters, I believe. 50, 60 meters, somewhere in there. And then we're planning to work our way up to around 2,000 meters. The main ridge is at around 2,000 to 2,300 or so meters. We're running a little behind on our waypoints, but we should make it to at least the deeper part of that ridge. Unless we end up seeing an amazing coral forest where we get stuck looking at everything. Hmm. Awesome. Thank you. We can take a quiver site. Well, no, I think we're gonna take the whole colony 'cause it's a black coral. So yeah. No point in trying to only take a piece of it. It looks like it's small enough to fit in a quiver, huh? Yeah. That's true. We haven't seen any fish in a while. We did when we first settled down. We saw like Synapha branchidae, some Acaridae, I think. Yeah, I can't remember what all we saw, but we saw at least 3 or 4 different species that I recall when we first settled down on the dive.
Yeah, we collect that one.
It's solid or gelatinous?
Gelatinous.
How did you collect it?
1 hour ago.
No, like how?
Private.
The net or?
No, we got it that part of the boat.
Yeah, that's what I'm saying. Like, if you grab it from gelatinous, wouldn't it break apart?
Yeah, we break up the silicone. We have the most important plate that is the delta.
Okay. Is it rare or is it something new or rare?
Yeah, we utilize something new.
Yeah.
Maybe it's a new report of this hobby, but maybe it's a collection also. Tauro, ¿qué cree que es Tauro? ¿Eric?
Podría ser, no sé, es poco raro. Yo no puedo ser, tiene ramificación raro, no sé. Seguro que es. Quizás con la vista de Glamour Shot podemos ver bien, mejor. I don't think it's Storopathes. I think it's growing maybe a little bit strange because of the way the currents are. Like it's growing more off one side than the other. It's weird. I don't know. Can we pan down just a little bit to look at the main part of the skeleton? Yeah, I'm at a loss, Maxie.
Me too.
Okay. It's a black coral. That's all we got right now. All right.
Yeah, the pattern is weird.
The branching pattern is weird. I don't know if it's like a response to where it settled or, yeah, interesting. Thank you.
Which weird?
9. It's a black coral, so it might be sticky. It might. Yeah, I think it's stuck on that one. You might have to hit the base against the inside of the to knock it loose. You might have to invert it to get it to go in. So weird. It's almost like that one branch has like decided to start a new colony. It's a weird pattern. Yeah, it is really interesting that it's like not balanced in how it's growing. That might just be a response to the environment because it was kind of nestled in against the rock. But we're not sure. Black corals are often quite sticky, so it's just stuck on the manipulator here. Oh, there you go. There you go. Yay. Looks like it was just stuck a little.
Samples done.
Okay, thank you. Awesome. Alright. Thanks. Onward we go when we're squared away. Hmm.
So the festivities you guys plan for Friday in the morning?
Send off. Yeah. Just yeah. We'll see.
That's if I wake up. I should be up.
Yeah, the decision is whether to stay up or whether to try to power nap and get back up again.
Yeah, I saw like Thomas sent out the thing that says you guys have something at 5:30. 5:30 a.m.
Mm. Alright. Working towards waypoint 7 when we're going to take a turn. I think that's another one of those colonies we just collected. It looks a little more even in its growth pattern. Some crinoids. Lovely. Keep seeing this small whip coral. We're seeing a part of the colony on the left, the crinoids. It's kind of going up these old lava flows. Starting to see less sediments. So hopefully that's a sign we'll see more diversity of corals as we move up this ridge and up these features towards the main ridge. Yeah, I think the black coral we collected was in good condition. I'm suspecting it has a weird growth form because of where it had settled. And it was trying to take advantage of the currents. We just passed one other colony that looked a little more balanced. So if we find another colony, we'll probably give it a zoom. The last one we passed wasn't in a great location for us to zoom in on. And a nice glass sponge coming up. I think. Actually, yeah. Yeah, it's a glass sponge. We saw one of these earlier. A larger one. This one is partially dead. A little slime star. At least I think it's a hymenaster, right?
Yep. Slime star.
Salty.
Yep.
Why does it look different than the other?
This is very soft and good.
Awesome. That's good. Thank you. Mm. Yeah, slime star. It's a common name for that star that we just passed. Yeah, slime star in the genus Hymenaster. It's in the family Pterastidae. Pterasteridae, rather. Still heading up an area with these kind of cliffs or walls. So just moving up slowly to make sure the ROV is safe and doesn't get caught. And then we kind of have this old lava flow that's been broken up. Little coral whips that we've been seeing still. If you look kind of on the edges of a lot of these rocks, you'll see them. Still seeing a lot of sponge skeletons. Ooh, this one is a tall sponge. This might be a selfie stick to see its perspective on how tall it is relative to Sebastian.
Yeah.
Look at that.
Wow, nice.
Let's get some lasers on it so we get the size before we do that, and then you can turn off the lasers. Yeah, the lasers are still a few, a little bit below. There we go. Cool. There we go. Awesome. 10, 20, some 30 centimeters. 25, 30 centimeters across. Yeah, nice. Thank you. We're gonna bring out the selfie stick and show you the size of this sponge relative to the size of Sebastian. The sponge is a Caulophacus.
Okay.
So how tall is Sebastian? 2 meters? So let's see. So Sebastian right now is around just over a meter off the seafloor. So we're about just 2 meters off the seafloor right now. Getting up to 2.5. So just to give you some perspective, Sebastian is around 2 meters tall, and we are currently 2.2 meters off. So yeah, this is 2.5, 3 meters. Yeah, you can turn off the lasers now if you want. So you can get your glamour shots. I don't think so. I think we're good. I think we have a really what's the word, optimistic goal in terms of making it to the top of the ridge. So yeah, it would be a cool one to get a 3D one for sure. Plenty of room to work around it. I think we can essentially do the same thing, just taking the pictures later instead of doing a snap. Kind of looks with all the particles being blown off, it almost looks a little bit like it's steaming. Yeah, my best guess right now is it's somewhere between 2.5 and 3 meters tall. It's definitely taller than Sebastian because we had to raise up. It might even be 3.5 meters tall. I have to kind of look at the images later, but Sebastian's about a meter off. Yeah, I would say it's like around 3 meters. Ooh, someone's hanging out in the back. Yeah, no. We're not going to try to sample that. We're going to leave this ancient sponge. And whoever's made a home in it. Awesome, that was cool. we go. I'm not sure. I don't know enough about how fast sponges grow to give you an age estimate there. I don't know if Maxi knows. Maxi, ¿qué edad piensas que tiene?
1,000.
Yeah, Maxi's guess is like 1,000 or so, but yeah. Yeah, my best guess is around 9 feet tall until we can actually sit there and look at the images with the lasers and do some measurements.
What is that?
Are the lasers back on?
It's another sponge with the different
Awesome, thank you.
meters are attached around the stop.
Yeah, it's a sponge stock, yeah. Phonophore? Oh, we made it to waypoint 7. Let me drop that down. Yay. I know, we're actually moving at the pace of like an hour waypoint. We're supposed to be moving at a pace around 30 minutes. Every once in a while we go 30.
30 minutes away.
Yeah, we're a little optimistic on our plan for the day. So maybe here in this flatter area we can go just a little bit faster. Made the turn towards waypoint 8. Let's see. For the next little bit, I think it's a little bit flatter for the next few waypoints, and then we'll go a little downhill. We still have a ways before we go up more of a continuous uphill on the ridge. This area has lots of ups and downs. It's an interesting ridge feature that we're going up from a geomorphology standpoint. Like that sponge base, yeah.
Like a tectonic cap or something.
Yeah.
Yep.
Maybe a passport or something.
You can see every once in a while these sponge stalk bases and dead stalks. There's another good size sponge coming up. I think this one we can just kind of fly by. Yeah, you can pop lasers on it. Yeah. Since we did a nice zoom on one of these earlier. Yeah, nice. Yeah, we're back to seeing more sediment again. You kind of see the like ripples and flows of the sediment. Must be some pretty decent current in this area to have some of these ripples. And I would say these sponges that we're passing now are more like a meter or so tall and a half a meter or a little less, less or more in width. at their widest point. So yeah, probably a meter, meter and a half tall for these. When we get our lasers on it, it's 10 centimeters apart, so yeah, these are looking so 10, 20, 30. So from this angle, it's a good 40 centimeters or so across. So it's probably a little under a meter tall, maybe half a meter wide. I think we can go a touch faster for this bit here with the ripples. See if we can make up a little bit of time. The sea cucumber off on the left. Yeah, it's nice. This lone rock here. You can kind of see how the flow is, it's like altering the flow, and you get all the sediment built up behind it and all the little rock pieces. You know, I imagine there's some sounds down there, that some of the animals make some sounds, but I don't know if anyone's done soundscape work at these depths. We are currently sitting on a ship, and we just send down the remotely operated vehicle, and we're just viewing it live from the ship. It's a nice sponge there. But, you know, the animals, as they move around and do different things, are going to produce sound. Even if it's not a way that they're using for communication, sounds are produced. We have Juliana coming in for Rich.
Yep.
Back to another wall here. It's like a bamboo skeleton there, caught on the base of a sponge skeleton. Triangles. Yeah. Yeah, I think it's also kind of quiet in an area where there's fewer fauna, fewer animals. So right around waypoint 8, where we're coming up to now, is a local high. We'll go through a little bit of a valley again after waypoint 8 as we head towards waypoint 9. It's kind of like this up and down stretch that we're going to be going through before we have the more consistent upslope from about waypoint 14 or so. So we are coming up on waypoint 8, so the next few are kind of up and down. So you'll kind of see us getting closer to the seafloor, and then as we move down slope, probably a little further away. We will make another turn here at waypoint 8 as well. It's kind of like a little zigzag to try to stay on some of these features as this ridge, as we head towards the main part, the main local high of the summit on this ridge. Yeah, we can like mid-water again. Yeah. So we're going to do a little bit of mid-water so we don't have to try to work down this feature backwards or sideways. And it'll allow us to catch up a little bit on our time as well as we come down from this feature before we start heading back up. It's a little, I'd say, a short downslope before we start going up the next feature between this waypoint and the next. Can you take a look at what is over there on this right here?
Yeah.
Actually. Yeah, I don't think it's too far before we get to the low. Just trying to see which coral this is. It's like a coral. By a sponge stock, and I'm not sure from here if it's like a chrysogorgia-like or if it's a black coral. It's looking to me like it might be a chrysogorgid. Yeah, it's a chrysogorgid. It's a good sized one. Yeah, we do a quick zoom. It's nice. And you can take the lasers off too if you want. There you go. Yeah, it's in focus. Alright. Whatever the lighting is making that sponge look like it's gold. Luxury lighting. Awesome. Can you take a look at what's on that the oaf? Little, is that like a gastropod or something on it? Where is it? Huh, that's weird. Hey, what?
You don't get a good.
Yeah, that's what I think too. Well, now that I've seen that, can we also zoom in right here? Oh, that's a squatty. Yeah. Okay. Can we pop the lasers back on and see how big this piece is here?
Oh, he beat me.
I see one. I see the other 10 centimeters. So I think we could try to go ahead and take a sample like, right below that squatty. How big would that be? Would that be quiver size, or is that too big? Or it would go, or we could put it in bio box 4. Yeah, let's go ahead and collect that. I feel like we can get like a 4 for 1 right here, so.
Ooh, bonus.
I like
Yeah.
that lots.
Yeah.
Yeah.
Alright, I'll forward for you then. Popping. That's how long it's gonna be, right? What's that? Lock is on the right. Lock is on the right. I'm gonna bring it to your left. Alright. So we rotate around. So we ram up. No, it's not much. Oh, okay. Just hold it forward. Ready?
Yeah.
One branch of that reservoir here?
Yeah. Yeah, vamos a corregirlo con el part con el off que tiene como algo como un caracol sobre el off, además el squatty. Vamos a intentar por 4. So, bueno.
4 and 1.
Yeah.
Done? Alright. Get this portion right here for you.
Yeah, it's very possible that the stock is a Calophacus remnant. That's one of the corals we've seen. We've also seen some Bolosomas, so it could be a Bolosoma remnant as well. Both of those are stocked. So I would say it's more than likely one of those 2 types of sponge stocks. There's also an anemone down below there. What? Yeah, I think this is the stalk that that piece is attached to. So I think right where that anemone is. Yeah, you can cut just below the anemone, and I think it'll bring that stalk with this piece with it.
Below the anemone?
Yeah, I think just right in that. I can't tell for sure from here, but I think just below it. I think it's on that. I think that's the branch it's on. If a bigger piece comes, we'll find somewhere else to put it. Yeah, we'll find a place. It'll work out.
What's up?
I think so. Oh yeah, there it is, right there. Yeah, that's
Maybe just outside the blade.
I think if you twist it, it might just come oh, maybe not. There it goes. Yeah. This one usually will break pretty easily, even if you don't cut it. Yeah, it is. I had a feeling it would be. But it looks like it'll fit in the quad, huh?
Yeah.
Okay. Yeah, let's do a glamour. Alright. We got a 5 for 1 with the anemone, I think. Beautiful. And the hidden anemone. Awesome.
Super zoom.
No, I think we're good with those. Yeah.
Alright.
Let's pop this.
Watch out flare there, Mr. Chong.
4. Try for a quad and 4 if possible. If it's too long, we can go for 2B, but I'd prefer the.
It's huge.
Is it huge? All right, we can do for 2B then.
2B?
Yeah. 2B.
You're good then. I can get in that quad for sure. Yeah.
No.
Challenge?
No, let's.
Or you want the quad or you want to get
Let's just put it in the BB2B. Let's get it in there. Make sure we don't lose our squatty and our bonus samples. I'll take the larger sample for the 5 for 1. Did it jump ship?
It's in there.
It's in there. Oh, I can see it. Yeah, it's in there. Awesome. That's fine. Alright, then onward we go. Well, I guess we're doing shift change right now, so
Just because teams have changed. Yeah, I don't see the time.
okay. Sample is logged. BBTV. Price Agorda.
It's all up to date.
There's Squatty. Oh,
Started
Moss. And an enemy.
saddle, get water in there.
So Mike and Sunny are coming in for Ben and Tyler. Thank you both.
That's the last shift to the cruise, Ben Hur.
Oh, Ben, where?
Oh, we go all the way around. Oh. Does your shoulder hurt?
No,
It was their last piloting shift of the cruise. So it'll be a night of goodbyes. Alright, thanks. We'll just continue along the transect with lasers on.
From her, say goodbye. But are you, so you, let's say you're going to bed on?
No, we still have 2 hours. Brunch shift.
So you're not going to be here?
No more flying. Such is life.
Yeah.
And then we were just kind of
Kicking off.
mid-watering it, so trying to work our way backwards so you can go a little faster down slopes. The bottom's not too far off though, we could see it. Alright, and then Mari's also going to be coming in for me. She's just getting mic'd up here. They're going to do some stuff. So, alright. She's ready. I'll move my stuff over. Yeah.
Okay, I'm also going to take this opportunity to swap over to part 2 for the stream. If you're on YouTube, we will be redirected. Facebook, you'll be live in a few minutes. We'll see you guys on part 2.